All righty. Good. Hope you guys had a good weekend. Today is Monday, June 14th, 2021. We are on to our second week of summer, so it's got a ton of work to do today and really this whole week. We are going to finish up our discussion of cells today and then move up finally to our level where we can start talking about tissues. Again, I think tissues are the part of this section that is probably the most challenging. Just because it's going to involve a lot of histology, a lot of microscope work, and it's kind of stuff that people aren't as familiar with. Most of us, at some point in our middle school years made, that candy cell model where we've heard of a mitochondria before. You can know what the femoral region is. So this is really where we get to the stuff where people have some challenges with it, but luckily we've got some things that hopefully will help you to be successful with it. We've got a ton of assignments due this week. Your first PhysioX is due tomorrow. Hopefully you've started out already. It's not hard, but it's time consuming because again, there are 5 different labs you have to go through, collecting 5 different sets of data, collecting and answering questions for all 5 of them, and then producing 5 lab reports, all of which you'll turn in. Thursday, your Unit 5 review is due, as is a handout on the biointeractory skin color handout. That is an activity that you'll be doing online. Friday, you have your Unit 6 review due. And one of the main things we're going to talk about in lab today is going to be those epithelial tissue and connective tissue handouts that I want you to do. Again, as I mentioned, you're not going to be graded on your drawing skills, but you do need to put time and effort into them. They do need to be labeled. If these are not labeled, then you will not get full credit for them. And then there's also a fun fingerprint activity due. Then you get a whole weekend to study, and we come back for our lab and lecture exam on Monday, the 21st. You get a good 5-6 hours of break, and then we're going to dive right into the new material. Hopefully at this point in time, you figured out it is always important to be here and be active in the learning process. But in particular, it's going to be important to be here on Tuesday. On Tuesday, we're going to be forming our skeletal groups where you guys are going to be working together in groups to do bone presentations. So you'll be presenting bones and bone features, and we'll talk about that more obviously next week when we get to it. But I want to warn you and remind you that, again, it's always important to be here, but it's especially going to be important to be here on Tuesday so that you get put into a group. It's going to be doing 2 group presentations. And if you're not here, you won't get put in a group. And if you don't get put in a group, you won't be presenting. And if you're not presenting, you're not getting points. This is a graded activity. Very low key. I know I say presentation and people freak out. It is a very low key presentation. No one's going to be looking at you. It's going to be okay. You'll survive. But it is important to be here to be a part of that. All right. Questions on any of that? Excellent. Stunned silences. You guys know how much I love those. All righty. That is the game plan. Let's go ahead and get started. As I mentioned, we are into our last major cellular physiological process, and that is the process of protein synthesis. We've kind of hinted at this and the importance of this several times already during this class, but now we'll actually finally get a chance to talk about the specifics of how this takes place. As we talked about and know, it is our DNA that contains the information for making proteins. And there's this genetic concept that one gene produces one protein. Again, the genes, as we talked about, are in your DNA, and they basically determine everything about you, from your hair color to your skin color, to your eye color, to your blood type, to your predisposition of heart disease, to if you're part of the proud 11% that are left-handed. All of those types of things, those characteristics are determined by your genes, and we have thousands of them. And like I said, in genetics, there's this concept of basically one gene produces one protein. However, as we learn from the structural organization of proteins, we know that there are those 4 levels of the functional organization, structural organization of proteins. And almost all proteins are comprised of multiple subunits put together. So in reality, it's more appropriate to say that one gene produces one polypeptide, one string of amino acids, which we then put together to make proteins. Multiple polypeptides are put together to make one protein. So again, a little modification of that general rule, but it's all basically the same thing. Now, to make proteins basically requires a two-step process. The first is a process called transcription, right? As we've now learned the anatomy of the cell, we know that the DNA is located where?
Inside the nucleus.
Inside the nucleus. And where do we make polypeptides? Where do we make proteins?
In ribosomes.
Yeah, the ribosomes. And where are almost all of the ribosomes located?
The rough endoplasmic reticulum.
Rough endoplasmic reticulum, which is out in the cytoplasm. So we have this issue where we have the information in one location and where we need to make the proteins located in another. Right. So what we need to do is make a portable and a temporary copy of this information. Right. It's very, very similar to in ancient times. Right. If we went back to medieval times, knowledge was power. Right. Books, information was incredibly, incredibly important, incredibly vital information. And so it would be locked up in places like castles or locked up in places like churches. And could you just show up with your library card and check out a book if you wanted to back in those medieval times?
No.
No, if you needed information, you could go and ask for it. And if they deemed you worthy, they would provide you with a bit of that information, but they wouldn't give you the whole book. They basically would make a copy of the information you want, and then you could leave with that, right? And who were the people who made the copies of that information?
Transcribers.
Yeah, well, they were the scribes. Exactly. The scribes were the transcribers. And actually, again, as we talked about, knowledge was so important. Knowledge was power. Ironically, most of the scribes were illiterate. Basically, scribes would look at a piece of paper and see what was written on it, not having any idea what it was. And their job was just to copy it basically letter by letter. They had no idea what these words meant because if they could read, right, they would have access to that information and that information could get out. So ironically, most of the scribes were illiterate and their job was to just make a copy of the small bit of information that you wanted so that it could leave, but the main stuff could stay there. Now, if you were to go to medieval times and go up to that castle and they deemed you worthy and gave you a sheet of paper with the information that you wanted on it, would it do most of us here in this classroom any good? Would that bit of information, that piece of paper, do you any good, do you think? Would it really provide you the information you needed?
No.
Well, the answer is obviously no, because I'm asking the question, but why not? Well, what was the language that most of those important knowledge books were written in back in the days?
Latin.
Latin. How many people in here speak Latin? Can read Latin? Anyone?
Big Latin.
Not quite the same thing. Close, but not quite the same thing. No. So if you really needed to use that information, you were able to go back in time, you were able to get that copy. What do you still need to have done with that piece of paper?
Translated.
Yeah, exactly. And that is the second step in this process. The second step in this process is translation. We have to change the language. Right? In transcription, we go from the permanent temporary version of the information to a movable temporary version of that information, DNA to RNA. But notice the language is still nucleic acids. So we're making a copy in a dialect, you could argue, of it, but it's still the same language. However, what we need to do is change the language of that information. We have to take it from the language of nucleic acids to the language of proteins. And proteins are made up of amino acids, right? And as we talked about already, that occurs in the ribosomes. So transcription occurs in the nucleus. Translation occurs at the rough endoplasmic reticulum, basically where we have most of the ribosomes. And so to make a protein is really a two-step process: transcription and then translation. Now, again, let's talk about transcription first, and we need to talk a little bit more about the information we talked about a bit in mitosis, talk a little bit more about DNA. Again, we are making a temporary and portable copy, and that's really the big key. It is temporary and portable copy of the genetic material. It's able to leave the nucleus, but it's not supposed to last forever. And again, it's using the same language, nucleic acids. This is going to work in an incredibly similar fashion to what we saw in DNA replication. Remember, in DNA replication, basically, we had two strands of complementary base pairs. So again, we'll do this with a real simple one, C, G, T, A, C, G, right? If that was one strand, what would the complementary strand to that be?
GCATGC.
There you go, right? Using those complementary base pair rules, we were able to make a copy. And remember, in DNA replication, basically what we did is we separated those from each other. Once we separated those from each other, we could then use new nucleotides to come in here and make a new copy. For one strand, use the template for a copy of the second strand. And what was originally one strand of DNA now becomes two strands of DNA, right? That was our DNA replication. And of course, remember, we had dozens of enzymes that were involved in that process. But remember, the primary two that we talked about was the helicase that unwinded the DNA, and then the DNA polymerase, which made the copies just like we did. So that was how DNA replication works. Well, something similar occurs, and I'm going to cheat here. Get rid of that. Something similar happens here in RNA polymerase. We again have, and let's even use the same strand, TACG. We have that same strand of DNA that we had before. And this one may be the one that contains the gene. This is the one that is the gene. So if this is the one that is the gene, this is what we call our template strand. We know it's going to have a complementary strand to the DNA, and we've already talked about CG, AT, GC. And this is what is known, as either the complementary strand, as we know, or it is also sometimes referred to as the coding strand. And since I went ahead and made this smaller, let's go ahead and do that. So the information we need is just on one copy of the DNA. Now, again, we are going to need to pry the two strands apart so we can make the copy of the information that we need. However, the difference here in transcription is once we've made the copy, we're going to let the DNA go back together again. So unwinding it, reading it, and then letting it fold back together is all a singular process. And as such, there is one primary, again, dozens of enzymes involved in this process, but the primary one involved in unwinding and making the copy is what we call RNA polymerase. So DNA polymerase allows us to make copies of the DNA strand in deoxyribonucleic acid. RNA polymerase allows us to make one temporary portable strand of the DNA, but using RNA polymerases. And that's the other thing we need to remind ourselves about as well. There are two big differences between DNA and RNA. In DNA, obviously, it has the deoxyribose is the sugar. whereas in RNA, the sugar is ribose. So that's obviously one of the big differences. But the other big difference between DNA and RNA are the bases. Remember, in DNA, the bases we used were A, C, G, and T, adenine, guanine, cytosine, and thymine. And remember, we saw how we have these complementary base pair rules. One of the purines, thymine in this case, adenine in this case, can form 2 hydrogen bonds. One of the pyrimidines, thymine, can produce 2 hydrogen bonds. So those are the complementary base pairs. The purine, guanine, is able to produce 3 hydrogen bonds. The pyrimidine, cytosine, is able to produce 3 hydrogen bonds. So those are the complementary base pairs. But if you remember way back when we were first talking about nucleic acids, we said that there were 5 total bases. And that's because RNA uses A, C, G, and U. For RNA bases, they use both guanine and cytosine. So they are going to form complementary base pairs with each other. However, for RNA, we use a different pyrimidine. That different pyrimidine is uracil. Now notice it is very, very similar to thymine, which is why it's also able to form 2 hydrogen bonds, and it will bind with adenine. So basically anywhere where in DNA we would use thymine, in RNA we are going to use uracil. All right. Questions on that?
Nope.
Excellent. Let's come to the whiteboard here. So let's take a piece of DNA. Oops, that should all be capitals. Excellent. And as I mentioned, this one is what we will call the template. And it is the template because, again, this is the one that it contains the information we need. This is the gene. Now, obviously, genes are much, much longer than this, but you get the idea. This is the gene, the information that we need to make the protein that we want to make. Okay. Now, as we also mentioned, we are going to have a coding strand. And in this case, what would our coding strand be?
GC, TA, TG, CA, GT, GC, ATGA.
Excellent. I'm going to assume that's right. I didn't bother looking, but I figure these things are pretty easy. And again, this is, as we mentioned, this is either 2 ways we could think of this. This is the complementary strand, which again just means that it may have a different gene with different information on it, but we don't care. Or it is also what is known as the coding strand. Either of those are fine ways of defining our complementary strand using this complementary base pair rules. All right. So essentially what happens during transcription is using that RNA polymerase, we are going to temporarily pull these 2 strands apart. And when we temporarily pull these 2 strands apart, now using RNA nucleotides, we are going to make a temporary movable copy of this information. So in this case, what would our RNA polymerase using complementary base pair rules be?
G, C, U, A, U, G, C, A, G, U, T, no, G,
That's all right.
C, A, U, G, A.
Perfect. Absolutely perfect. All right. We would make this copy using RNA nucleotides. It would then be able to leave the nucleus. And once it left the nucleus, our DNA would come back together and would be completely unchanged by this process. And at its core, that is exactly how this process works. It really is almost that simple. Now, is it ever almost this simple? No, of course not. There's got to be a little bit more to it, and of course, more vocabulary. But at its core, that's really all we're doing. All we're doing is what we did in DNA replication, peeling apart the DNA, using our complementary base pair rules, using the special enzyme RNA polymerase to make a temporary copy. But not surprisingly, there's a little bit more to the process than this. So let's talk about that. Now that we basically have a simple understanding of what's going to happen, let's talk about some of the logistics of what's going on. Starting with the first major issue that we have. Now, again, most of you are too young to probably remember this very well, but when we first were able to, get the Human Genome Project was about reading the DNA of humans and figuring out our genetic information, because that was going to be able to tell us so much amazing stuff about humans and how they function. And prior to the Human Genome Project, it was believed, just based on the average size of a gene and the amount of DNA that is inside of a cell's nucleus, that humans would have somewhere on the order of hundreds of thousands of genes that would control the things that are going on in the body. And one of the most remarkable things that came out of the Human Genome Project is the astoundingly small number of genes we actually have. Instead of having somewhere on the orders of hundreds of thousands, we only have tens of thousands of genes. In fact, it at the time was realized that only about 1.5%, why is that not writing, of the DNA in our nucleus codes for proteins. Only about 1.5% of our DNA, and again, let's not be careful, is our genes. Let's say that. There is a massive amount, which at the time was called junk DNA, that we thought were just these weird, random, repetitive patterns of DNA that were found in there that didn't appear to have any type of function. What's interesting since then, one of the hugest areas of research that is going on is this field of study known as epigenetics. And epigenetics is actually looking at this junk DNA, this overlying massive amounts of DNA, and the potential implications. And there appears to be some potential function for these repetitive patterns of DNA. It's not just random, it's not just junk, but that's a whole completely different field of study. For us, what we care about is the fact that still only about 1.5% of the DNA are genes that code for proteins. And that gives us our first big issue. If the job of the cell is to make a protein, and I were to just randomly take a dart and throw it at your DNA, what are the chances that I'm going to hit a gene, let alone the gene for the protein I want to make?
Very, very slim.
Yeah, it's very low. It's almost impossible. So what happens is rather than just randomly going in there to grab a gene and hope for the best, we need a way to prompt that RNA polymerase to go, okay, here is where you start. And so the first major step in our transcription is a process that is known as initiation. With initiation, we have to have a way, we need to tell the RNA polymerase where to bind to start reading. And luckily, we have something that does that. We have an area that we call the promoter region. Promoter region is basically a special sequence of DNA nucleotides that tells the RNA polymerase, bind here and start. So if we cheat and bring back up my DNA, here at the beginning of my DNA, and we'll make it pink because that's fun. There may be, what is that promoter region? And it's something like AAA, TTT, or some type of special sequence of nucleotides that says, all right, RNA polymerase, this is the gene we want to read. This is where you need to come and bind. So come here, grab onto this, and start your reading there. So that is basically step one in transcription. We need to start the process. Then that RNA polymerase is going to come and do exactly what we said it was going to do. It is going to temporarily peel apart the two strands of DNA using our complementary base pair rules. It is going to come down and form that. And heck, since I already did it, I'll just go ahead and cheat and do this. And it's going to go down the line making the copies. This is step two in the process, and it's what we call elongation. Elongation, just like we talked about, as the name indicates, is where our RNA polymerase moves one base at a time and using complementary base pair rules, adds a new RNA nucleotide to a growing strand. And again, remember, one of the big keys in that process is it uses uracils instead of thymines. Now, once this continues down the line, once it grabs onto that chromatin, that DNA, is it basically just going to keep going all the way down till it gets to the end of that chromosome?
No.
No. So eventually, we need to tell it to stop. And the way we tell it to stop or to terminate is to basically reach a termination sequence. And again, this will be some sequence of amino acids, something like AT, 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 or something like along those lines. and that basically tells the RNA polymerase to let go and stop. At which point, we now basically have two things. Our DNA right? Our DNA folds back up and is completely unchanged by this process. All right, so it's exactly the same thing as it was. However, what we have now is a new single strand of RNA nucleotides. This new strand of RNA nucleotides then needs to be modified. Basically, sections will be cut out. It'll be pasted back together. And when done, we have a single strand of RNA nucleotides that contains the information we need to send to the rough endoplasmic reticulum to do a job. And what do you think we call this, dare I say, message that needs to go to the rough endoplasmic reticulum to tell it what to do?
Translation.
Well, it's going
mRNA.
to take place in translation, right? And
mRNA.
mRNA would get you partial credit on the exam, but remember, we don't allow abbreviations. So what does the M of mRNA stand for?
Messenger.
There you go. All right, so we have our messenger ribonucleic acid. Let's write it all out. There you go. At the end of this process, we have our messenger ribonucleic acid, which once you wrote it out once, you can abbreviate, typically lowercase m, capital RNA. So you could also argue, I guess, that this is 4 steps to this process, because once we get that strand of RNA, we have to modify it. And this is just that simply transcription. Now, there's 1 or 2 more teeny vocabulary things we need to add to this, but at its whole, this is transcription. So we've done it here on the whiteboard. Let's take a look at the pretty pictures from your textbook and talk about this process. Again, there are dozens of enzymes involved in this process. In fact, we already know this. RNA is the primary, but there are many more. Right? Lots of other enzymes, energy molecules, nucleic acids, nucleotides. In fact, there is so much stuff wrapped around the DNA where this transcription is taking place that we can actually see this dense cluster of stuff under a light microscope. And what would we call this dense cluster of stuff where transcription is taking place inside of our nucleus?
A nucleoli?
Exactly. Remember, we talked about that nucleoli. Those nucleoli are seen under the light microscope. It shows that protein synthesis is taking place. That nucleoli is basically the RNA polymerase and all the other things tightly packed around the DNA where it is taking place. So where you see those nucleoli, that is where transcription is taking place inside of our nucleus. Right, we had already talked about that. We'd already mentioned that, but see, we're putting pieces together. I like that. Excellent, excellent. So, as I mentioned, with only 1.5% of our DNA coding for genes, coding for proteins, we need to find the right place to bind. So here we have our RNA polymerase looking for that promoter region. Again, it's going to be a special sequence of nucleotides like AAA, TTT, or something along those lines. And our RNA polymerase will bind to that region. Notice we have 2 strands of DNA, right? Our template strand and our coding strand. But remember, we only are going to be reading one. We only care about one of these, the template strand. The template strand is the one we're going to be reading to make our information. So our RNA polymerase binds on, pulls the 2 strands apart. Again, it's only hydrogen bonds holding it together, so that's pretty easy for it to do. And then just reading the template strand, it is going to, using those base pair rules, start to make a copy of it using RNA nucleotides. Now, that does make this area right here pretty darn interesting, because what we have is a double strand of nucleic acids where one strand is DNA and one strand is RNA. And as we know, anatomists love to name everything, so they happen to call this region the DNA RNA hybrid region. So during elongation, just like we've done numerous times, our RNA polymerase moves down the strand of DNA, reading just one copy and using those complementary base pairs and RNA nucleotides to make a growing list. And again, this is the one thing I don't like about your textbook. Again, technically, it is not messenger RNA yet, right? Basically, what we are getting here is a single strand of RNA nucleotides. Remember, it has to be modified before it can become messenger RNA. So we get this growing strand. And again, this is a pretty simplistic illustration. Let's look at a lot better one. Here we go. Notice here is our RNA polymerase. Notice at one end, it is unwinding the DNA. At the other end, it is winding it back up again. So the DNA is completely unchanged by this process. But notice it is only reading the template strand. It's only using the complementary base pair rules on our template strand. So if it sees an A, it puts a U. If it sees a G, it puts a C, and so on and so forth. If it sees a T, it puts an A, and so on and so forth. So notice, and again, they've colored this for us to remind us purple is that deoxyribose with those deoxyribose sugars for our sugar backbone, whereas the blue for the RNA represents that it's the ribose sugar. So different sugars and the one nucleotide that is different, the uracil instead of thymine. And again, still not messenger RNA yet. It doesn't read down the whole strand of DNA. Eventually it reaches a termination signal like AT. And when it reaches that special base sequence, it lets go of the DNA. Notice our DNA is completely unchanged by this process. Our RNA strand is completed, but remember, it must be processed. And that processing occurs in the nucleus. And once that processing has occurred, cut and spliced and modified, then and only then do we have our completed strand of messenger ribonucleic acid. And even though we've already said it a couple times, once you have that completed messenger RNA, where does that completed messenger RNA go?
To the rough endoplasmic reticulum.
Yeah, to the ribosomes, most of which are at the rough endoplasmic reticulum. And how does it get there?
Vesicles?
No, vesicle is a good guess, but nope, we don't need a vesicle. If only our nucleus had some openings that something like a
The
messenger.
pores?
There you go, those nuclear pores, exactly. Our messenger RNA is able to sneak out those nuclear pores. And remember, the rough endoplasmic reticulum is right around the outside of the nucleus. So it ends up straight at the rough endoplasmic reticulum, where it can find a happy little ribosome to read it and do step 2 in this process, translation. Excellent. That is transcription. Questions on transcription? All right, excellent. The good news is
I actually had a quick question.
yeah, go ahead.
I don't know if this is just asking a dumb question, but
The only dumb question is the question not asked.
when the DNA strands are going through the RNA polymerase and it's breaking those hydrogen bonds, is it like dehydration and hydrolysis? Like,
No, that's a great question. No. Dehydration synthesis and hydrolysis are used to break covalent bonds, right? Hydrogen bonds, being much weaker, can actually just be pulled apart. So there doesn't require to, again, remember, hydrogen bonds are the weakest types of bonds. They're not sharing electrons. It's basically just electrical forces, positive and negative charges that are kind of holding these things together. So it's just a matter of separating them from each other. It doesn't require any kind of chemical reaction to do that. That's a great question. Yeah, no, it's just merely a mechanical force to pull them apart because they're so weak. Excellent. Any other questions on this? All right, so like I said, protein synthesis is a 2-step process. We've got step 1 under our belts now, and that is definitely a good thing. The bad news is translation is a little bit more complicated. So let's
actually, sorry, Professor, would you be able to go back to your white?
Oh, yeah, sure.
Take it.
Yep. All right, excellent. Any other questions or anything else from anyone before we move on? All right, we have then our messenger RNA. And that messenger RNA can now lead us to translation that takes place in our, let's go back to the whiteboard actually. In our rough endoplasmic reticulum. So again, translation. Translation is going to occur in the rough endoplasmic reticulum. But remember, as we talked about, the key to translation is this is where we change the language. And to do this, we have an issue, right? How many letters are there in the English alphabet?
26.
26. And we take those 26 letters and arrange them into different formats, and those give us all of the words we need. How many letters in the Nucleic acid alphabet.
5.
True, but remember in this case, we're dealing with just RNA, right? Because of our messenger RNA. So technically, how many do we have in our RNA alphabet?
4.
4. And if our goal is to change it into amino acids, how many different amino acids do we have? I know you told you didn't have to memorize all the different names of all the different types, but anybody remember how many different amino acids we actually have?
20.
20.
20. So how does that work? How do we go from an alphabet with 4 letters to an alphabet of 20 letters? Can one, like A, stand for 5 different amino acids that we can just use interchangeably? Can we
No.
just interchangeably switch amino acids in a string of polypeptides?
No.
No. Remember we talked about conditions like cystic fibrosis, or not cystic fibrosis, sorry, sickle cell anemia. With sickle cell anemia, the change of one amino acid is sufficient to weaken that hemoglobin to a point where it can become damaged in a low pressure environment and cause all the problems that go along with that. So how are we going to do this?
By switching up the sequence of the RNA alphabet, you can get 24
Right.
total.
And that's the key. We need not just one letter, but we need a sequence of nucleic acids to code for an amino acid. Right? Obviously, if you just used 4, and that would be 4 to the first power, like A, B, C, or where's my up arrow thingy? There it is. 4 to the first power, that would only give us a total of 4 options, like A and C and U and G, and that wouldn't be enough. But what if we did 4 to the second power? Right, so we did like AA or AU or CG. How many is 4 to the second power? How many different options would we have there?
16.
16.
16, would that be enough?
No.
No. But if we went 4 to the third power, how many do we get then?
64.
64, right? So things like AAA or AUA or CCC or so on and so forth. We can get 64 different combinations, which is more than enough for what we need. And so that's what we have to do. We have to take that strand of messenger RNA and break it down into these sequences of 3 nucleotides. That will code for a single amino acid. And what do we call this special code sequence of 3 nucleotides?
Codons.
Yeah, we call it a codon. See how convenient that is? So these are our codons. Excellent. So the first thing we have to do in this process of translation is change the way we read the information from just single letters to codons. Any textbook worth its salt has a chart like this that shows this process. So see, for instance, your first one is a C, your second one is a C, and your third is a U, so CCU. Notice codes for the amino acid proline. Now notice 64 is more than the 20 we need. So not surprisingly, as you look on this list, there is a fair amount of redundancy. So notice there are 4 different codons that can code for proline. And do you need to necessarily memorize these 4 codons that code for proline?
No.
No, you don't have to do that. You need to be able to read a chart like this, but you don't have to memorize that. However, as I do look at this chart of codons, I do see that our artist has highlighted 4 of them. One of them, they have highlighted in green. And what does green usually stand for?
Start.
Well, green means go, but you've got
Go.
absolutely the right idea. This green one here is indeed what is known as the start codon. And guess what you do at the start codon?
Promoter.
Right, this is where you start translation. And when you start translation, there is one and only one amino acid you will use. So not only is AUG our start codon, but it also codes for a special amino acid, methionine. So while you do need to memorize this entire chart, you absolutely positively have to memorize that start codon, know it's the start codon, and know that it also codes for methionine. But I also noticed that there are 3 other color-coded codons. What are they?
To stop.
Yeah, these are our stop codons. And as you can see, they are UAA, UAG, and UGA. And as the name would indicate, this tells us to stop translation. Now, do these happen to code for an amino acid as well?
No.
No. So any of these 3 can be used to tell the process of translation to stop. So while you do not have to memorize this entire chart, these 4 codons you absolutely positively need to memorize. Know them, know what they do. All right? Questions on that?
Nope.
Excellent. Now, as I mentioned, translation is a little bit more of an elaborate process. One of the things we absolutely positively must have is our messenger RNA. But messenger RNA is just one of the things we need to be successful in the process of translation. All right, again, we're converting the message from nucleic acids to amino acids. But there are 2 other important RNAs that we need for this as well. Obviously, we have to have the messenger RNA, but we also have to have transfer RNA and ribosomal RNA. So let's briefly talk about those as well. Let's start first with transfer RNA. Transfer RNA, as it turns out, is by far the smallest of the RNAs that we are going to use in this process. It is a single strand of nucleotides that is only about 80 nucleotides in length. When we look at the secondary structure of this, here, let's actually do this first. You can see its secondary structure is this kind of 3-leaf clover shape. Very distinct. That folds up in this slightly more complicated 3-dimensional structure. But typically, when we draw this, notice they've drawn one over here symbolically. I find it even easier to simply just draw it as a straight rectangle. But whether you bend your rectangle or it's a straight rectangle, whether you look at the 3-dimensional structure or most easily at the 2-dimensional structure, you will see that there are 2 very important regions to this transfer RNA. The first of those very important regions is up here at the top, and this is our amino acid binding site. Up here at the top is a little twig comprised of a sequence of nucleic acids that is able to bind an amino acid. And let's be more specific, a specific amino acid to bring it to the party of translation. Now, for simplicity, because there's only one you've had to learn so far, let's say for argument's sake that this one happened to be methionine. This transfer RNA is the one that brings methionine to the party. Now, lucky for us, we happen to know, because we have to memorize it, the specific sequence of RNA, of messenger RNA that codes for methionine. And what was that special sequence of messenger RNA that codes for methionine?
AUG.
AUG, excellent. So down here on our messenger RNA, we would have something that said AUG and then whatever the next ones were, and so on and so forth. So let's give us a little bit of space so that we don't get too confused. So here's my messenger RNA with that AUG codon. Now, we need a way for this transfer RNA to know that it needs to bring methionine to the party. And so while this is our codons, at the base of our transfer RNA, we have something we call the anticodon. And that anticodon uses our complementary base pairs So it's going to use complementary base pair rules to match the codon on the messenger RNA. So if I happen to have a transfer RNA that has methionine bound to it and needs to bind to my start codon, what would the appropriate anticodon be? What would the complementary base pairs to AUG be?
TAC.
But it's messenger RNA, so it would be, it's RNA. So it wouldn't be T.
UAC.
UAC.
UAC. Excellent. So notice there are two important points on our transfer RNA. There is the very specific 3-sequence code at the bottom of our cylinder, known as the anticodon, that will allow it to bind to our messenger RNA. And there is the very important binding site at the top, where a specific amino acid would be held. Now, conversely, and let's go ahead and make sure we understand this. And I won't bother with the symbol this way, if I had an anticodon that happened to be, in this case, UUG, right? What would the codon of the messenger RNA be in that particular case?
AAC.
And if that was the case, would we know what amino acid that our transfer RNA was holding? Now, again, do we necessarily have to have it memorized?
No.
No, but could we figure it out? Yeah, let's cheat and go back. I've kind of made a mess of it, but let's see if we can find it. So the codon is AAC. So I find A, I find A, I find C, and it's ASN, whatever the heck that is. All right. Did I have to have that memorized? No. But if I had a chart like this, could I figure out exactly what amino acid that was? Yes. And that's the point. Our messenger RNA is going to have these two key components to it, a specific amino acid on one side and a specific anticodon at the other side. And it's going to be a very important molecule necessary for translation. All right.
So I just want to make sure I understand this correctly. So the mRNA would connect to the anticodon and the amino acid associated with it would be waiting at the attachment site.
Correct.
Okay.
Yep. So yes. So basically what's going to happen is our transfer RNAs are going to line up by their anticodons, matching up to the codons, and bring the right sequence of amino acids there. And then we need to put those together. Now you've hit on a huge key. These things are great by themselves, but we need a place to arrange them, to align them, and we need a place for our chemical reactions to take place. Well, you guys already know what those are. Those things that are going to line them up and allow the chemical reactions to take place are the ribosomes. We already know the ribosomes are going to be responsible for making the proteins. So it is the ribosomes that are going to make those. And not surprisingly, ribosomes are made up of two things. Those two things are proteins and ribosomal RNA. Now, the molecular composition of this is far more complicated, so I'm not going to make you recognize it the same way we do the transfer RNA. But instead, we'll draw our ribosomes globularly. So let's cheat and go back to our whiteboard. So again, we know our ribosomes are made of proteins and ribosomal RNA. And ribosomes have two subunits with very technical names. One is made up of fewer proteins in ribosomal RNA. One is made up of more. So these are known as the small and large subunits. The small subunit, as the name would indicate, is smaller. See how easy that is? And the small subunit has one important feature to it. It has a binding site for the messenger RNA. So we'll draw this as a little black dot. Where it's going to bind onto our strand. Of messenger RNA. Our large subunit is, of course, large. And the large subunit actually has three, well, 2.5 for the transfer RNA. One of these is what is known as the A site. One of these is known as the P site. And one of these is known as the E site. Now, as we know, transfer RNAs bring and carry an amino acid. That is why the A site is called the A site, because this is where a transfer RNA brings in a new amino acid. once we start putting amino acids together in a strand, what do we call a strand of amino acids?
Polypeptide.
Polypeptide. Guess what the P stands for? Not your question.
Polypeptide.
It contains the strands of polypeptides. And guess what the transfer RNA does in the E site?
Exit.
There you go. That's why I kind of called it only a half a binding site, because the transfer RNA doesn't really stay bound in the E site. It just uses the E site to escape. So E for exit, P for polypeptide, A for amino acid. They made it pretty simple for us. So again, a new transfer RNA brings a new amino acid into the A site. The transfer RNA in the P site contains the strand of polypeptides, and then in the E site, the transfer RNA leaves. And essentially, this is what a ribosome is. And while I've done a truly amazing job of drawing this, we also have the pretty symbolic structure from our illustration from our textbook as well. So again, the ribosome brings the messenger RNA and the transfer RNA together, and it also facilitates the chemical reactions that are going to take place as well. Many of the proteins that are part of the ribosome are enzymes that make the chemical reactions occur faster. As I mentioned, they have two binding sites, an A site and a P site. And then we also have the E site, which is the exit site. So there's the small subunit and our messenger RNA binding site, the large subunit with the A site, the P site, and the E site. All right. Questions on this?
Nope.
Excellent. So now we have all the pieces. Now we have all the players. What we can do now is put this process together and describe the process of translation. Now, as I mentioned, translation is a little bit more complicated than transcription. So I think it'll be best if we come at this fresh. Like I said before, we'll go through it 2 times. The first time I will do it on the whiteboard. For that, I want you to just watch, listen, think, observe, not bother writing, and then we'll go through the whole thing again with all the pretty pictures and all the pretty words and make sure that it makes sense to you. But let's go ahead and take our first break first. It is 9:18, so that means we'll come back at 9:33, take a good 15-minute break, come back fresh, come back caffeinated, and we will restart at 9:33. And I will start the recording at that point. All right, any questions?
Nope.
All right, see you guys in 15 minutes. Like I said, let's go ahead and go through this process once just here on the whiteboard. Again, for this, I encourage you just put your pens down, watch, think, listen, interact with me, answer my questions that I ask you, and think about the things that I'm saying to try to make some sense of it. All right, so we started out with our bit of messenger RNA, which we have here. That has left the nucleus out that nuclear pore and gotten here to the rough endoplasmic reticulum or even one of the free-floating ribosomes. And so basically the first thing that has to happen is 4 things need to come together. Obviously, the messenger RNA is the first thing that needs to come together. But also we need the small subunit of our ribosome, our large subunit of our ribosome, and our first transfer RNA. We need all of those things to come together, and we need a good, oh, I don't know, dare I say, starting point for those things to come together. What might a good starting point for those 4 things to come together to be? If only there was something that would help us to start this process of translation.
Methionine.
AUG.
Right. How about the start codon? Absolutely. You absolutely have the right idea. We are looking for the start codon. And what is the start codon?
AUG.
AUG. So notice what's going to happen is our small subunit is going to start moving down. Stop it. There we go. Our messenger RNA, to lo and behold, it finds our start unit, UAG. And using that binding site, which I didn't bother to draw because it was too complicated, it is going to bind to our messenger RNA at that start codon. So the first thing we're looking for, and let's go ahead and highlight it because it's useful to do so, our start codon. Excellent. And once the small subunit binds to it, our large subunit is going to bind to it as well. And again, remember, as we know, it's not going to be as easy to move these the same way all the time, but I think I can make this work. We know that there is the E site. There is the P site. And there is the A site. I think we can make that work. There we go. Something like that. Excellent. So that's thing 3 that comes together. And thing 4 is going to be our first transfer RNA. Now, can any old transfer RNA come in here and bind to this site?
No.
No, I'm asking the question, so the obvious answer is no. But it needs to be a transfer RNA that has what specific aspect to it?
Complementary.
Yeah, the complementary anticodon. Excellent. And what would the complementary anticodon be in this case?
UAC.
Excellent. Did I go too big with this? Nope, I didn't. Okay, just looks bigger. Excellent. So the transfer RNA, let's go ahead and change the color of it just to make sure we understand that. The specific transfer RNA with the specific anticodon is going to come in. And in doing so, it is going to bring a specific amino acid. And as we know, what is the amino acid that is going to be brought in by our first transfer RNA?
TYR.
Not TYR. What was the what the start codon is? Not just the start codon, but what amino acid does it code for?
Methionine.
Methionine, exactly. Methionine, which we will abbreviate appropriately as MET. Excellent. So the first thing that has to happen is these 4 things need to come together. Our messenger RNA, the small subunit of our ribosome, our large subunit of our ribosome, and our first transfer RNA. Now, once this comes in, notice we have an A site that is open. And so the next thing that happens is a new transfer RNA is going to come into the A site. Of course, this transfer RNA must have a specific anticodon. And what would the specific anticodon here be?
GGC.
Right, so not any old transfer RNA could come in here, but only the one with the specific anticodon to our codon, right? It is the anticodon that must match the codon of the messenger RNA. doesn't need to be that big. That is going to come in and bind. And of course, with it, it is going to bring a specific amino acid. Now, if we cared, could we actually go back to that chart and figure out which amino acid it would be?
Yeah.
Yes.
Yeah, we could, but quite frankly, we don't care. So we'll just put amino acid 2 here because it's the second one. All right, like I said, you don't have to have them memorized. You need to know the first one memorized. You need to know it's methionine, but the rest of them you don't have memorized. So we can just put amino acid 2. We could figure it out if we wanted to, but we don't have to. All right. Notice at this point, we have our ribosome locked and loaded with as much stuff as we possibly can. And now that we have 2 transfer RNAs in here, it is now time for our chemical reactions to take place. And I do mean chemical reactions, plural, because what's going to happen now is 2 things. The first thing we are going to do is we are going to make a bond. And then once we make a bond, the second thing we are going to do is break a bond. So that is the short term of what we're doing, but let's work our way through this. What is the process that we've learned? What is the name for the process by which we make bonds between molecules?
Hydrogenation.
Close. You're right in that we have those 2 hydroxyl groups that we're going to form together with an oxygen. And in that process, we bring out 2 hydrogens and an oxygen. And so we called that not hydrogenization, but close to that. What was it? Dehydration synthesis. Exactly. We're going to use dehydration synthesis. And when we use that dehydration synthesis, what we are going to do is we are going to form a peptide bond between the 2 amino acids. Okay? Again, remember, covalent bond between amino acids we call a peptide bond. And of course, when we do this dehydration synthesis, we get a water, because after all, that's what dehydration synthesis does. It gives us a water. And that water is going to be useful for breaking a bond, because what is the process we use to break a bond?
Hydrolysis.
Exactly. To break a bond, we're going to use hydrolysis. We're going to use the water we just made to break the bond between, oops, so I spelled between, right? The amino acid and the transfer RNA in the P site. So notice we make that bond. When we make that bond, as a result of that, we get a water molecule, which I'll just draw as a blue circle. And then we're going to use that blue circle to break the bond between that methionine and the transfer RNA in the P site. So now that we have 2 transfer RNAs locked and loaded into our ribosome, we can do our 2 important chemical reactions, making a bond and breaking a bond. We make the bond between the 2 amino acids, peptide bond, using dehydration synthesis, which gives us a water. We can then use that water for hydrolysis to break the bond between the first transfer RNA and the first amino acid. Questions on that?
You're probably going to go into it, but after that bond breaks, does that transfer RNA leave to another, get something else?
You're absolutely correct. That is exactly what we're going to talk about next. Now, at this point, all of our chemical reactions have taken place. There is nothing else for us to do. So, Friday night, you're sitting at home bored. So what do?
Go out, get your friends.
True, you could if you had friends. Or if you didn't have friends, you could be sitting there on your app, right? Again, there's a lot of sites you could have. I have a huge social media profile myself. My Parlor site is still down, but I still have my OnlyFans and my Grindr site. And when I get bored, what do I do there on my Grindr site? I swipe right. And that's exactly what my ribosome is going to do here. My ribosome swipes right. Now that there is nothing else left to do at this location, the entire ribosome shifts right down our messenger RNA. And notice when that happens, as Brian pointed out, our first transfer RNA is now in the E site. And what does it do in the E site?
Leaves. Exits.
Exits. Excellent. Notice our second transfer RNA is now in the P site. And again, remember P stands for polypeptide. It has the growing polypeptide strand in it. All right, so let's get rid of that. Well, just put that out of the way. And then a new transfer RNA can come into the A site. And of course, when that new one comes into the A site, can any old transfer RNA come into this A site?
No.
No, only the one that has the specific anticodon we need. And what would that anticodon be?
CUU.
CUU, excellent. There we go. Only the one that has the anticodon that matches the codon here. And of course, when it comes in, it is going to bring an amino acid with it. Do we happen to have memorized which amino acid that is?
No.
No. Could we figure it out if we cared to? Absolutely. Yep. But we don't care at this point, so we will just mark it as amino acid 3. Oops. Now that I have amino acid 3 here inside of this, I have it locked and loaded with all the transfer RNAs that I possibly could ever want. What do I need to do now?
Make a bond, break a bond.
There you go. Which one do I do first?
Dehydration synthesis.
Right. So which one is that's making the bond, right? So I'm going to make a bond between the second and the third amino acids, the amino acid in the P site and the amino acid in the A site, using, as was mentioned, dehydration synthesis, which just so conveniently is going to give me a water. And why is getting that water so darn useful?
To break the bond in the P site.
Exactly. I can use that water for hydrolysis to break the bond with the transfer RNA and the amino acid that is in the P site. I've done all the work I can do at this location, so what do I do next?
Swipe right.
Move to the right, exactly. So my large subunit moves to the right. My small subunit moves to the right. And notice as that happens, The transfer RNA that was in the P site is now in the E site, and so it is going to exit. Now, as someone mentioned, this can then go off and can get another amino acid put onto it, right? In fact, sometimes some of these transfer RNAs can be used dozens of times in the making of a single sequence of amino acids. Stop that. So
it's always gonna continually go out and get the same.
Right, because it has
amino
the
acid,
same
'cause it's
anticodon, it's always
codon.
going to pick up the same amino acid. So the one that has the UAC is always going to get methionine, right? And so they're always going to pick up their unique, their specific amino acid. All right. Now, again, because I'm not bothering to draw new ones, I'm just reusing my drawings here. But of course, now a new transfer RNA is going to come in. Can it be any old transfer RNA? Yeah, these questions should be getting easier because they're the same answers over and over again. Exactly. What transfer RNA's anticodon will match this one here?
AGA.
AGA. Excellent. It is, of course, going to bring some amino acid with it. Do we know which amino acid? No, we don't care. Could we figure it out if we wanted to? Yes, absolutely. But for understanding how translation works, we don't need to know what the amino acid is. We just need to know that it's the next one in the line. And so we'll call it amino acid 4. We are now locked and loaded and ready for some chemical reactions. So what happens next?
Dehydration synthesis.
Excellent. We make a bond using dehydration synthesis between amino acid in the P site and amino acid in the A site. Conveniently enough, that dehydration synthesis, oops, too big, gives us a water. And that water is useful because what can we use that water for?
Hydrolysis.
Hydrolysis, breaking a bond. And that's exactly what we're going to do. We're going to break the bond between the amino acid and the transfer RNA in the P site. And what happens next?
Keep moving to the right.
Keep moving to the right, exactly. This will happen thousands of times, right? A single polypeptide can be made up of hundreds to thousands of amino acids, and this keeps happening again and again and again and again and again, right? Form a bond, break a bond, shift to the right, bring in a new transfer RNA, form a bond, break a bond, shift to the right, and so on and so forth, thousands of times until finally something new and different happens. What is, whoops, hold on, let's cheat. Get that out of my way. What is that new different thing that's going to happen? There you go. Samuel's got it. Suddenly, we have a very unique and need to be memorized codon here, a stop codon. And someone again remind me what the stop codons were?
UAA. UAG.
UAG.
UGA.
UGA. Excellent. And is that one, and is this one of those?
Yep.
Excellent. So notice we have now reached our stop codon. And let's go ahead and highlight that. Here's where something different happens. Again, a transfer RNA is going to come in, and not any old transfer RNA. It again has to have the specific anticodon. And what would the specific anticodon be in this case?
ACU.
So again, it can only be a specific anticodon that is matchy-matchy to come in. But remember, when we were looking at our chart for the codons, unlike the start codon, which also coded for methionine, did this stop codon code for an amino acid?
No.
No. And that's because these transfer RNAs associated with a stop codon don't bring an amino acid to the party. Instead, they bring a water. And why might it be useful for these stop codon transfer RNAs to bring a water?
Hydrolysis.
That water will be used to break the bond between the amino acid in the A site and its transfer RNA. And once we do that, is there anything holding this together anymore? No. And so what's going to happen is everything comes apart. The large subunit moves away, the small subunit moves away, and what we're left with, oops, I should have moved those out of the way a while ago. What we're left with is basically a string of amino acids. Oops, that can stay there. This can move here. And what we're left with is our polypeptide. All right, the final result of translation is a string of amino acids, right? A polypeptide. And where is this polypeptide going to go?
Golgi.
Yeah. Goes to the Golgi apparatus or Golgi complex. Again, remember both of those are appropriate terms for it, where it is cut, folded, and added to subunits. To make a functional protein. There you go.
Professor, when it exits the when it exits right here, does it get wrapped up in the vesicle before heading to the Golgi? Or
it can, but it doesn't have to. Remember, there are vesicles that will carry the proteins to the Golgi apparatus, but also if this was a free-floating ribosome, then it wouldn't necessarily be able to be bound into a vesicle. So it'll still migrate to the Golgi apparatus, because remember the Golgi apparatus is right next to it, where it'll either be brought into the Golgi apparatus internally, or it'll bind to its surface and be modified from there. And notice one other key point on this that I want to make, because again, I think if you think about this, notice in this process, every single protein we make starts with the amino acid methionine. Does that mean that every single protein in your body, the very first amino acid is methionine?
yes.
No. When it is made this way, yes, absolutely. But remember, in the Golgi apparatus, we're going to cut off pieces of it so that it'll fold up the right way and become functional. So one of the parts that is cut off is usually the beginning part. So are there proteins that start with methionine in your body? Sure. But when they're fully functional, not everyone is. It is when we make them, but once it goes to the Golgi apparatus, usually the first thing the Golgi apparatus does is cut off the beginning of it so that it'll fold up properly. So yes, so not every functional protein in your body has methionine as its first amino acid, but when we're doing translation to make that sequence, we always start with methionine. All right. Notice this one's a little bit more elaborate than transcription, but not much, right? It's really basically a simple 4-step process, right? Lock and load with transfer RNAs, make a bond, break a bond, shift to the right, and continue the process, right? We have a starting point, we have an ending point, but other than that, it's a pretty simple repetitive process that goes around and around and around and around.
Now
All
with
right.
the RNA at the stop coding, would the next sequence be a start coding?
Great question. No. So notice also, you absolutely have the right idea. That messenger RNA isn't a book that you read cover to cover. And why does a book have a cover?
To protect the inside.
To protect the insides, exactly. And that's what happens here. Think of it this way. When I've done this, I make one sequence of amino acids that, if I'm lucky, becomes one protein. If I'm telling my cell to make proteins, do I probably just need one copy of that protein?
No.
No, I probably need hundreds or even thousands of them. Notice my messenger RNA is not damaged by this process. So a new ribosome could come and bind to it and start its process of moving down the line and then letting go. And while one ribosome is part way, a second one can bind on. In fact, your textbook's got this great picture. Using an electron microscope, it shows a single strand of messenger RNA that has like a dozen ribosomes all moving down, making lots and lots of copies. So we want to make lots and lots of copies. However, at the same time, once we start making a protein, do we want to be making that protein forever? No. So at the same time that this messenger RNA is floating around inside of the cell, there are these enzymes, and the enzymes will come and bite off the first nucleic acid. And then another one will come by and bite off the next one and the next one and the next one. Right? By having this padding, like the cover of the book, we have some protection. But eventually, are we going to get to a point where it breaks down that start codon and now this messenger RNA is no longer functional? Yeah. So that way, we're also not making proteins forever either. We want to make more than one, but we don't want to make them forever. So having buffer nucleic acids on both sides allow it to last for a while, but not last forever. Great questions. Any others?
Where's the transfer RNA just like pulling the free floating amino acids from?
Within the cytoplasm, right? Remember, in the cytoplasm, as we've talked about, and actually your book's got let's cheat. Your book has a great, I've got a very simplistic version of this we'll go through in a second, but your book has this great in focus where it talks about this. And remember, inside the cell, inside the cytoplasm is all sorts of stuff, ions, ATP, all sorts of other things. And one of the things that are freely floating around are amino acids. So when our transfer RNA loses its amino acid, it can actually find a special enzyme, and that special enzyme will take the correct amino acid and put it on top so that it can be used again. Like I said, some of these transfer RNAs can be used over a dozen times in the making of a single protein. So yeah, so these amino acids are just freely floating around inside of your interstitial fluid because you ate them for breakfast. They got into your blood. They got into your interstitial fluid. They diffused into the cell, or the cells actively pumped them in the cell so that they were all floating around. Right. But it's also why it's important to make sure you have a well-balanced full meal because, diet. Because if you're not getting a particular amino acid, some amino acids your body can produce, but there are about a dozen or so that your body is not capable of making. And so they have to come from your diet. So if you're not getting those amino acids from your diet, right, then you're going to be malnourished. You're going to have trouble making some proteins, and that could affect the functions of your cell, the functions of your body, and lead to all the problems associated with malnutrition. Now, it doesn't mean if you have one bad meal, because again, remember, a lot of it is stored in your liver, but weeks, months without getting that particular amino acid could ultimately cause problems. So again, I've done the best I can to describe this. Your book does an excellent job of describing this process in this in focus. So if anything that I said didn't make sense, I encourage you to go there and read it. I do have one other simple example. And again, notice this one is so simple that this is not from your textbook, but it's from another textbook, but I think it served the purpose. Notice their ribosome, they didn't even bother putting the exit site on it. They just have the two binding sites, the P site and the A site. But let's go through the process again now. So notice we have 4 things that need to come together. Our messenger RNA, our small subunit of the ribosome, our large subunit of the ribosome, and our transfer RNA. Those four things all need to come together. And they all come together on our start codon. And remember, the transfer RNA that comes in must have the appropriate anticodon for the codon. And since it's the start codon, we know this first amino acid must be methionine. Right? It's the only one we have to know. The rest we could look up if we wanted to, right? Or if you had to for an exam, but on an exam, I'd have to give you that table so that you would be able to determine what the appropriate amino acid would be. This is the only one you have to have memorized.
And this is all basically just a description of the primary level of protein synthesis.
Yes,
Sequence.
this is how you absolutely great point. Absolutely. Translation, you are 100% absolutely correct. Translation is really the process by which we make a single strand of amino acids. And you are absolutely correct. When we think of the structural organization of our proteins, this is the primary primary level, pardon me. Structural organization. Right? It then goes to the Golgi apparatus where it cuts it, allows it to fold up, forms the tertiary structure, puts it together with the subunits, and makes a functional protein. So yes, this makes that primary level of the structural organization just a single strand of amino acids. Great point. All right, so those four things all together come together along the start codon. But then we need our second transfer RNA to come in. Again, it can't be any old transfer RNA. It has to be the one with the correct anticodon for the codon that is there. And it's going to be bringing a specific amino acid, which if we cared enough, we could look up. Once it's locked and loaded in that A site, then again, remember, two chemical reactions are going to take place. We are going to first make a bond, forming that bond between the two amino acids, forming that peptide bond between the two amino acids, using dehydration synthesis, which is going to give us a water as a result of that. And once we get that water, what can we use that water to do?
Hydrolysis.
Hydrolysis, where we can break the bond between the first amino acid and our first transfer RNA. Once this is done, everything shifts to the right. Swipe right. Our first transfer RNA leaves out the exit site. Second is now in the P site because it's holding the growing polypeptide strand. And a new transfer RNA bringing a new amino acid comes into the A site, bringing the new amino acid. And this process continues hundreds, really for most proteins, thousands of times. I don't know what the average number of amino acids for a protein is, but most protein subunits are made up of thousands of amino acids. So this is happening thousands of times until something special happens. That something special is reaching that stop codon. Notice here in this case, the stop codon is UAA. And someone remind me what's special about the transfer RNAs for the stop codons?
They don't have an amino acid, they're bringing water.
Exactly. They do not bring an amino acid. They bring a water molecule for hydrolysis. And once it breaks that bond, everything comes apart. We now have our completed polypeptide strand, which goes to the Golgi apparatus, or the Golgi complex, either is fine. Our ribosomal subunits come apart, and notice our messenger RNA is unchanged by the process. So many ribosomes can all be reading it at the same time. Your books not only got this great image that shows it to you, but notice also here we see this image where, again, new ribosomes bind to the messenger RNA, move down, make their individual polypeptide sequences, let go at the end, and continue on. And while it's got this pretty picture from your textbook that shows it, your textbook also has this great electron microscopy view where you see inside of a cell a single strand of messenger RNA that has something like a dozen, 2, 4, 6, 8, 10, 12, more than a dozen ribosomes all moving down that messenger RNA, all making a single copy of those polypeptides to make the thousands of proteins that this cell needs.
So on that previous picture, it showed, like in the book and all the everything like that, it shows the ribosomes as a globular unit. It's really a top and a bottom, or are they just kind of floating around looking for a pair?
So remember, some ribosomes are free floating. However, some of them, the subunits, the different subunits are attached to the rough endoplasmic reticulum. So what'll happen?
So I'll say like the small and the large, are they small and the large just kind of floating around? Like they're not one unit.
Correct. They are floating around separate and for the free floating ones, but for some of them, some of those subunits are attached to the rough endoplasmic reticulum. So what happens is one subunit is bound to the rough endoplasmic reticulum, the messenger RNA comes in, and then the second subunit comes on and binds to it. So that's how we keep it locked in place. So no, there isn't the both in the center and it squeezes up. No, typically one subunit is bound and the other is free floating. All right, excellent. So notice here we have now talked about this process. We have gone from the strand of DNA, so a particular gene on our DNA, that template strand, converted it into messenger RNA with codons, used the anticodons of our transfer RNA to give us a sequence of amino acids. And notice, not only do we need to be able to describe the processes of translation, describe the processes of transcription, but this is a good point to bring this up. Being the smart, sophisticated students that you are, if we have two closely related processes, how many possible essay questions are there for that?
Two.
So definitely there's two. Describe process A, describe process B. Is there any other possible essay questions? Well, I'm asking the question, so what's the obvious answer?
Yes.
The obvious
Yes.
answer is yes.
Yes.
So what is the third possible essay question when you could have stuff like this?
Differences?
Yeah, comparing them. How are they similar? How are they different? Right? For instance, transcription stays in the same language, nucleic acid to nucleic acid, whereas translation changes the language from nucleic acids to amino acids, right? And that's one of the important things about, so much to talk about. That is one of the important things about this type of process. If you have a comparison type of question, that doesn't mean describe both processes. I think I've said it before in this class. If not, I will definitely say it now. For the most part, I do not care how you present me your information. I don't care if it's paragraphs. I don't care if it's bullet points. What I care about is that you provide me the information and that you are specific. Now, the one thing that I will warn you is often when people use bullet points to describe things, they don't go into the detail necessary. And remember, I'm going to be a stickler for detail. But the same thing is true for these comparison type of questions. If I ask you to compare translation and transcription, that doesn't mean that you should describe to me the entire process of transcription, and then you should describe to me the entire process of translation. That is not a comparison. So, as I said, while you are not required to do it this way, one of the ways that I find organizing these types of questions to make it a little bit easier to make sure I'm comparing them is by having your two columns, but then adding a third. And here the third, you can actually have this be like the characteristic. So for instance, I just mentioned how one of the differences between transcription and translation is the languages. Transcription, it stays the same, where it's nucleic acids to nucleic acids. And are you going to be abbreviating all of this on an essay question like I am?
No.
No.
No, but again, for speed, I'm doing it here. Changes from nucleic acids to amino acids, right? So by having that characteristic, it helps me to organize. What's another way that they are different from each other?
Location
I love it. Where do they occur? Where does transcription take place?
in the nucleus.
Where does translation take place?
Ribosomes.
Or we could say rough ER. And again, I'm abbreviating because of speed, not because don't do that on the exam. Excellent. Give me at least one more.
The end product.
Okay, excellent. I like that. End product. What is the end product of transcription?
The mRNA.
Yes. Excellent. Messenger RNA, which of course you're going to write out. What is the end product of translation?
Polypeptides.
Yeah. Excellent. Right? How they start. Transcription starts at a promoter region. Translation starts at the start codon. Right? There are dozens of ways you can do that. Now, again, and I'll likely ask for 3 or 4 or something along those lines. So you could just say all 4 things for transcription and say all 4 things for translation and get full credit for that. However, make sure you're comparing apples and apples, right? If you say transcription uses the same language of nucleic acid to nucleic acid and translation occurs at a ribosome, while you are giving me information, are you comparing those two things when you do that? No, right? And make sure you talk about both. Don't just say translation occurs at a ribosome and transcription doesn't, right? Okay, if it doesn't, that's great. Tell me where it occurs. It occurs in the nucleus. So make sure you're providing me with both pieces of information. This type of an organization is something that can help you to do that. It's not required, but for me, it makes it easier for me to organize to make sure that I'm actually doing that comparison between the two. Apples to apples and oranges to oranges and grapes to grapes. The last thing, and again, it's not the last time I'm going to say it, but the last point I want to emphasize on this is, again, when you have two related processes like this, read the questions carefully so you can answer them correctly. I say this over and over and over and over again. However, on every exam, there's at least going to be 1 or 2 people where they're going to read the essay question, and the essay question is going to be describe the process of transcription. And they're going to tell me how the messenger RNA leaves the nuclear envelope to go to the rough endoplasmic reticulum, where the small subunit of the ribosome binds to the start codon, and then the large one binds, and then a second transfer RNA comes in using those anticodons, and we make a bond, and we break a bond, and it shifts to the right, and yammer, 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 and all of that showing tremendous knowledge, tremendous content, tremendous information, tremendous detail. And what's the problem with all of that?
Not answering the question.
Yeah, I asked for transcription. You know, the irony of that is you answered a harder question. You clearly have information and knowledge, but the problem is I asked for transcription and you didn't give me transcription. In fact, the only thing about transcription you said is the starting point, that it started with messenger RNA, and transcription really ends with that product of making messenger RNA. So that may be a 10-point essay question where you gave me a massive amount of information and you're getting 1 point. Because while you showed me great knowledge, you didn't answer the question. Please read the questions carefully so you answer them correctly. If I'm asking for transcription, give me transcription. If I'm asking you for translation, give me translation. If I'm asking you for a comparison, don't describe both. Compare both. All right. Look, we spent 2+ hours talking about this. Are one of those 3 possible essay questions almost guaranteed to be on your exam?
Yes.
Yeah, absolutely. And do I know which one it's going to be?
No.
No, because they're all in a bank of questions together. You'll get one of those or something similar to them. Because here's the other side of it as well. That's the fun with this type of a question. Not only can we do what you guys just did. Oops, I want to go back to here instead. But if I gave you, for instance, a chart like this, whoops, or like this. If I gave you a chart like this, where I told you what transfer RNA anticodons associated with what amino acid, could I ask you to do this process in reverse? Could I give you a sequence of amino acids, and based on that sequence of amino acids, you would have to give me the template strand of DNA that built it?
For sure.
Yeah, absolutely. Yeah, exactly. So again, you need to know how, or I could do the other way around. If I gave you one of those charts, could I give you a sequence of DNA and ask you to give me the sequence of amino acids? Yeah, all of these are possible essay questions that I guarantee you are on the exam. Some of you will get each of these, right? And I have no problem telling you that because I know you still have to work on this. But we have one more major issue we have to talk about. Here is my problem. Your toenails, your eyeball, and your liver cells. Do they make the same proteins or do they make different proteins?
Different.
Different proteins. Do they have the same DNA or different DNA?
The same.
Yeah. Every single somatic cell in your body has the exact same DNA. But they do not make the same proteins. So not only is it make this protein, don't make this protein, but I need to be able to tell toenail cells what proteins to make and when to make them, tell liver cells what proteins to make and when to make them, and tell eyeball cells what proteins to make and when to make them. And how the heck do I do that? How do I tell all the different cells of the body which proteins to make and when to make them? How do we regulate this process?
Is it one of the communication systems?
Only we had organisms, organ systems that were specialized for communications. Absolutely. Do we have something like that?
The nervous or the endocrine?
Yeah, we have 2, nervous or endocrine. So what do you think? Do we have nerves that go to every single one of the trillions of cells in our body?
No.
So then how do we regulate this process?
Endocrine.
Endocrine. And what is it that the endocrine system produces to regulate this process?
Hormones.
Hormones. Producing and releasing hormones tells our cells what proteins to make and when to make them. Hormones are how we tell, hormones and other chemical signals tell our cells what proteins to make and when to make them. And that's how we can get toenail cells to make toenail proteins, liver cells to make liver proteins, eyeball cells to make eyeball proteins, all when they have the same DNA. All right. Questions on that? Awesome. Excellent. With that, then, we are done with cells. So we are done with everything we need to talk about from the cell level. And now things get interesting. We need to move up to the tissue level of organization. However, now our vocabulary gets a little bit more challenging. Now our comfort level is going to get a little bit less. So again, I think it is best that we come at this fresh with this information. So let's go ahead and take our second break. We'll take another 15-minute break. That means coming back at, looks like 10, what is it now? 27, so 10:42. And at, oops, that's not a 10. 10:42, we will restart. And at that point, I will start the recording again. If you have not done so already, of course, I'm going to change the screens now, and then I'll have to write this again. So let's copy that. If you have not done so already, then what I strongly encourage you to do is to print out your epithelial tissue and connective tissue handouts. So again, they're in the modules. So during this break is a perfect opportunity to do that. There are two handouts here. You see them here. This is the epithelial handout. And then there is also the connective tissue handout. These handouts are two important things. The first important thing is a graded assignment that you must complete. But the other equally, if not more important thing is that these are going to be your study guides for the lab exam. Again, there's a lot of information mashed together into this exam, but my guess is this epithelial and connective tissue probably is going to make up about 25 to 30% of your next lab exam. These two handouts. Easily 25%, if not more, of your first lab exam. And anything that important, could some of it bleed over into your lecture exam? Could I ask you to identify and describe the different types of cartilages in the body, or what are the main characteristics of epithelial tissues or something like that? Absolutely. So this is vitally important information. If you haven't printed it out yet, well, first and foremost, shame on you. But secondly, do that during the break so that we can talk about these when we come back. We're going to switch into lab gear. We're going to talk about tissues, introduce the concept of tissues, make some tissues, and then we'll talk about these handouts to help you to be successful moving forward from here. All right, questions on any of that? All right, I yammered a bit, so let's go ahead. And again, I want you to have a full 15-minute break. It looks like it's 10:30 now. So let's go ahead and resume at 10:45. So we'll restart at 10:45, and I will see you guys in 15 minutes. All right, any questions? All right, see you guys in 15 minutes. So we are done with cells, and we are now up to the tissue level of organization. And of course, as we talked about, tissues are comprised of cells. There are 4 different major types of tissues in the body. Can anybody identify what they are for me, or give me one at least?
Connective.
Connective tissues, excellent. And with a name like connective tissues, what do you think connective tissues do?
Connect.
Yeah, building is tall because it is tall. Absolutely, yeah. Some of their functions are to bind or hold things together, as the name would indicate with a connective tissue. However, connective tissues are also the most diverse. So therefore, both in structure and therefore in function. Can someone give me an example of a connective tissue?
Ligaments.
All right, ligaments is an example. Give me another one.
Tendons.
Tendons and ligaments are actually the same type of connective tissue. Excellent. What else? Guess what? Bone is a connective tissue. Guess what else is a connective tissue? Blood. Right? Are there two substances in the body more different than blood and bone? Probably not. Right? One is obviously a liquid, and the other is this crystalline hard matrix. Obviously, they can be very, very different in their structure, and therefore, of course, very different in their functions as well. So we talked about that bone allows for movement, allows for protection, allows for structure, blood for transportation, and so on and so forth. Excellent. Give me another type of tissue.
Nervous
Epithelial.
tissue.
Nervous tissue, I like that. Nervous tissue is a highly specialized tissue. Specialized for what?
Communication.
How does it communicate?
Electricity.
Yeah, a big electrical signal. Anyone know the fancy name? I think we talked about this once before, but I'll ask it again. Anyone know the big fancy name for the big fancy electrical signal that our nervous tissue produces?
Action potential.
Yeah, there you go. So it is specialized for communication, producing this big electrical signal we call action potentials. Give me another one.
Muscle tissue.
Another highly specialized tissue.
Is cartilage a tissue?
Cartilage is actually part of connective tissues. So yeah, connective tissues include things like cartilage, bone, blood, tendons and ligaments, all sorts of stuff like that. Absolutely. So it is definitely a tissue, but it falls under the connective tissue categories. All right, muscle tissue is also highly specialized, and specialized for what?
Motor, like moving around, transport.
Okay, movement. I like movement. Movement works, absolutely. Right, it produces contractions. And as we also mentioned, muscle tissue comes in three varieties, three flavors, three main types. What were the three main types of muscle tissue that we identified already?
Skeletal.
Smooth.
Smooth
Cardiac. And smooth.
and cardiac. Excellent. Excellent, excellent. All right, one more tissue type.
Epithelial.
There you go. Oops. Excellent. What do epithelial tissues do for us?
Skin.
Okay, skin is one of the locations where you find epithelial tissues, and you've got the right idea. Epithelial tissues line both the external, like skin, but also the internal surfaces of the body. And if it's lining those surfaces, obviously it's forming barriers. Right. And again, as we've talked about, are all the barriers in the body completely impenetrable?
No.
No.
No. So it also allows,
Microbial.
yeah, it allows movement in and out of the body. And in fact, epithelial tissues do something else important for us. They actually form our glands. So we use epithelial tissues to form our glands. Excellent. So there you go. We have our four main types of tissues. If I ask this on the lab exam or the lecture exam, right, identify the four types of tissues, this would be your answers: connective tissue, epithelial tissue, nervous tissue, and muscle tissue, and some generic descriptions of them. I do this because while you absolutely positively need to know this bit of information for the exam, as I also mentioned, nervous tissue is highly specialized. So we're actually going to wait till we talk about the nervous system to get more in depth on nervous tissue. And muscle tissue is highly specialized. So we're going to wait till we talk about our muscular system before we talk about our muscle tissue in depth. So while those are useful pieces of information for this exam, we are primarily going to focus on connective tissues and epithelial tissues. So for this first exam, you need to know there are 4 tissues in the body. You need to know what those 4 tissues are. You need to know a general description of those. But our focus for this exam is going to be the epithelial tissues and the connective tissues. Okay, so we need to know a little something about nervous and muscle tissue, but trust me, when we get to the muscle system and the nervous system, you'll learn everything you wanted to know and more about those tissues. And so again, here we see epithelial tissues line the surfaces, both inside and outside, form our ducts and glands. Connective tissues, again, most diverse, protect, support, bind, store energy, transport, provides immunity. And then our muscle tissue and nervous tissue. Now, the other, and here's a pretty picture from your textbook that shows this as well. The other important piece of information you have to remember is, as we said, tissues are made up of cells. So we need ways of putting cells together to make tissues. One of the primary ways we do that are with structures called cell junctions. Now, cell junctions are actually talked about mostly in the cells section, so we're going back to that a little bit, but since we're talking about tissues, here's a more appropriate place to talk about it. And as you can see from this pretty illustration, there are basically 3 main types of cell junctions that are formed in our cells to make our tissues. So this is an awesome picture, but you know how much I love to draw. So let's go ahead and do our own drawing of this. And of course, here is cell number 1, and here is cell number 2. And let's talk about the 3. Actually, let's do it this way. I'll move this over here and this over here, so we can talk about our 3 main types of cell junctions. The first of our 3 main types of cell junctions are what are called tight junctions, or also what are called occluding junctions. These are basically formed by long lines of interlocking transmembrane proteins. I need enough room to write all this, so make that a little smaller. So remember, a transmembrane protein is a protein that goes through the plasma membrane. Globular protein on one side, globular protein on the other side, and they basically interlock with each other. And a tight junction really isn't any more sophisticated than that. However, right, I think of them as kind of like teeth, like in a zipper, right? You have the 2 teeth from the zipper that lock together and hold them together. However, if you had a jacket, would you just put 1 tooth on one side and 1 tooth on the other and hope for the best of that jacket being held together?
No, you had multi.
No, and so there are massive long lines of these. The picture from your textbook does a really great job of showing this, and I'll just put 3 or 4 of them here. But the other thing that it kind of always has reminded me of is of like the quilting. Let's cheat and go back to the picture from your textbook real quick. Notice down here, we see rows and rows and rows. So again, your grandma, like mine, probably is part of a quilting club where they get together and they have the 2 big cloths that they've done all the decorations for. They put the batting in between them, and they all sit around in a circle and do these elaborate, delicate stitches to make these very fancy designs by basically smushing the batting of the quilt together in the center and this quilting process. So it's like a big zipper, it's like a big quilt over long surfaces. This is important because not only do these tight junctions help to give structure and shape to the tissues by holding the cells together, but the other important thing that these tight junctions form is by forming these big, huge mesh-like systems, they basically can limit the movement of water between the cells. As we know, water really likes to go where water really likes to go. So it is important to limit water's ability to be able to sneak from one side to another, right? Like in an epithelial tissue, right? How many people here have bathed before?
Yes.
Couple, excellent. For those of you who haven't, I highly recommend it, right? How many people here have washed dishes in the kitchen sink before?
Yes.
Yep.
A couple of you. If you haven't, I highly recommend that too. When you do the dishes in the sink, you have a sponge that you put in the water. And when you put that sponge in the water of the kitchen sink, what happens?
Soaks it up.
It soaks it up and it doubles in size. Is that what happened when you took a bath?
No.
When you stepped into the bathtub, did you suck up half the water and suddenly you swelled up like a big Stay Puft Marshmallow Man?
Nope.
No, and the reason for that is not surprisingly, skin is one of the areas where you find a large number of these tight junctions, limiting the ability of not just water to get in, but water to get out as well. Where else might tight junctions be really useful in limiting the ability of water to move into or out of our body?
Intestinal or digestive?
Digestive system, absolutely. We want to control what gets absorbed. Where else? Give me one more.
The blood vessels?
Yeah, whole cardiovascular system, absolutely. Do you want your blood vessels being all leaky, having water pouring out of them at all sorts of parts of your body? No, absolutely not. That would cause edema. That would definitely be a bad thing. How about your urinary system? The whole point of your kidneys is to filter bad stuff out of your blood. Once you filter it out and it's sitting there in your bladder, do you want all that crap being absorbed back into your body?
No.
No, absolutely not. So being able to have these tight or occluding junctions is very important, not just for limiting the movement and maintaining the shape and the structure of our tissues, but also limiting the movement of water. So very, very important that way. All right, questions on that one? All right, let's talk about a second type of junction. That second type of junction is called a desmosome. Desmosomes are much more elaborate in their shape. Let's take a look at the components. Actually, let's do it this way. There are really three main parts. The first of those parts is a peripheral membrane protein. Remember, peripheral membrane proteins are associated with the plasma membrane, but not actually connected to them. These are known as the plaques. They are very dense proteins, dense peripheral membrane proteins. That basically act as an anchor. Attached to these, and I'll draw it, are these transmembrane proteins that pass through the plasma membrane and have a globular component on the other end. So there are these transmembrane proteins. Here, let's do it this way. Plaques. These transmembrane proteins, there are numerous different ones, but one of the most common is a protein called cadherin. with a name like cadherin, what do you think cadherin does?
Adheres.
It adheres, absolutely. But what's interesting about these cadherins is they don't adhere all the time. Something special has to be present, something, dare I say, wonky, to make these cadherins sticky so they attach to each other. And guess what that wonky thing is that makes these sticky so they attach to each other?
Calcium.
Yeah, absolutely. So in the presence of calcium, they become these sticky transmembrane proteins that attach to each other, helping to hold the two cells together. However, if you also remember when we were talking about our cytoskeleton, we mentioned that one of the cytoskeletal structures are the intermediate filaments. We mentioned these are the ones that are the I-beams that help to maintain the structure, help to maintain the integrity of the cell. And one of the ways they do that is by attaching and anchoring these plaques in place. But if you remember, I also mentioned these transmembrane proteins actually have the ability to travel from one cell to the next, forming this big elaborate mesh. Notice between the plaques, the cadherins, and the intermediate filaments, we have this huge, massive bundle of proteins that are holding this whole thing together. These desmosomes produce these huge anchors to hold the cells together. Right? And again, if you think about it, this kind of makes sense. If I have two big, huge pieces of flat metal that I want to attach to each other, do I melt the entire surface of one of these pieces of metal to attach it to the entire surface of the other piece of metal?
No.
No, typically you put 2 or 3 or 4 little small spot welds. Those spot welds are really, really strong anchors at just one location that holds the whole thing together. And that's basically what these desmosomes are. These desmosomes are basically our spot welds, helping to hold the tissues together to keep the cells from tearing apart. Where might something like this be really useful?
Skin.
Yeah, once again, when I rub the surface of the skin or I itch the surface of my skin, right, does it come peeling off as a result of that?
No.
No. In fact, if you think about it, if I were to damage my skin by being out in the sun too long this weekend, I could damage and destroy those cells with that sunburn. And as a result of that, does each individual cell just die and flake off as a result of that? No, even dead and damaged, those desmosomes hold the cells together so that it peels off as a whole sheet of skin. So that peeling that occurs because we have these massive spot welds holding the cells together. Where else do we have a lot of force, a lot of pressure that it might be really important to hold all these cells together?
Bones.
Bone might be a good guess, absolutely. That wasn't the one I was thinking of. I was thinking of something a little bit more dynamic, back to the cardiovascular system. How about the heart? The heart is pumping, producing massive amounts of pressure. We need to hold those cells together. So back in our cardiovascular system, and the heart is a great place where you'd find these desmosomes.
Lungs.
Parts of the lung, maybe, but you got to be careful. Down here where the gas exchange takes place, the cells are really, really delicate. So we're not going to necessarily hold them together in that same way. Right?
I was thinking like the outside, the
True. Yeah, closer to the surface, absolutely. But they're not producing the pressure that the heart is. Heart is producing a massive amount of pressure, right? Any good samurai movie worth its salt, at some point, the guy's head gets chopped off. And if it's a good movie, you see that huge squirt of blood that goes 10 feet into the air from that massive force of the blood pumping out of the heart before their body's even aware of the fact that they're dead. So again, we need that strong force to hold them together. Excellent. The last type of cell junction is what is known as a gap junction. Now let's again talk about the anatomy of this. With a gap junction, what we actually have are these transmembrane proteins. Now let's draw them. That go through the plasma membrane, and they basically form a circular row. They attach to the transmembrane proteins on the other side. These special transmembrane proteins are called connexons. And basically what they form is a fluid-filled tunnel between the cells. Now, is this fluid-filled tunnel doing a great job of really holding these cells together tightly?
No.
No. So what are the function of these gap junctions? What's the advantage of having these gap junctions between the cells?
To allow more movement between them.
Yeah, absolutely. Jacob, what were you saying?
An exchange of proteins.
Right. Okay. So you guys have the right idea. By having a fluid-filled tunnel, what is allowed is things like proteins or ions or things like that can move from cell to cell. Basically, what this allows is for rapid communication between the cells. When there's one change in one cell, then that change affects the other cell as well. The other way we could think of this is it basically hardwires the cells together. And when it hardwires the cells together, they basically work as one. Where might this type of communication be important?
Nervous.
Nervous is a good guess, but and
Right.
there are some in the nervous system, but not as many as you would think. One of the places where you see a lot of these is the heart. We need the heart to work together as a single unit, as a single pump, so we want to coordinate that activity. And there is some in the nervous system, but not as much as you would expect, right? Because think of your house. Your house is hardwired. So there's a light switch on your wall. And when you click that white switch up, the light goes on. And when you click it down, the light goes off. And you could sit there for 15 minutes going back and forth and back and forth and back and forth and back and forth. And is anything different ever going to happen? Are you ever
No.
going to flip the switch and your garage door goes up as a result of it?
No.
No.
No, right? Because again, they're hardwired. There's no decision making in that. And that's the problem with the nervous system, because in the nervous system, we want to be able to make decisions. If I sit here on your Zoom camera with my mug of coffee in my hand, let's call it coffee, my mug in my hand, and I look you dead in the eye and I say, I'm going to kill you. Or a bear with an axe walks in your room right now, and that bear with an axe looks you in the eye and says, I'm going to kill you. You are getting the exact same stimulus from both of us. Are you going to respond the same way to both of us?
No.
No, absolutely not. So while hardwiring some things together can be useful, surprisingly in our nervous system, there's not as much as you would think, because we want to be able to modify our behavior. We want to be able to make decisions. When things are hardwired together, there's no decision making in that. There are times where that's useful, But in our nervous system, we want to be able to think. We want to be able to modify. We want to be able to make decisions. So there's not quite as much of it as you would suspect.
System.
Maybe in your muscles.
Jacob, I see you starting to talk, but I think you need to turn on your mic a little bit before you start talking, because I think by the time you finish talking is when your mic finally kicks in.
Yeah, I don't get this whole microphone thing. What about the muscular system?
So yes, muscular system, especially smooth muscle, like your stomach, things like that, that is definitely one of the areas where you're going to see it. Our cardiac muscle, when again, we want our whole heart to work together as one pump, as one unit. Those are places where you see it a lot more, absolutely, in muscle. There is some in the nervous, don't get me wrong. The nervous is the right answer, but not nearly as much as you would think. All right, I've done a crummy job of doing the drawings of these. Let's take a look at the pretty pictures and go through these again real quickly. Here we see that nicer image of the tight occluding junctions, two big, huge flat surfaces of our plasma membranes held together like a zipper, like a quilt, with those transmembrane proteins producing that watertight seal. And like we said, your skin, your digestive system, your bladder, all important places where you would find this. Here, we see a really nice illustration of those desmosomes. Notice we have those transmembrane proteins that are the plaques, not attached to the plasma membrane, but associated with it. Embedded within them are those integral membrane proteins, really transmembrane proteins, that are the cadherins attaching from the two sides. And notice also we have those intermediate filaments that your book does a nice job of showing us how they anchor the plaques in place. But remember, as we also talked about, these intermediate filaments can also actually cross from cell to cell as well, forming that big, huge, strong spot weld, right? Resists that separation, maintains the shape of the tissue, like in our skin, like in our cardiac muscle. And lastly, here we see those connexons, how they form a fluid-filled tunnel between the cells that we call a gap junction, allowing rapid communication between the two cells, hardwiring the cells together so they work together in one. And you find those in places like muscle tissue, especially cardiac muscle, and in a lot of smooth muscle, and also some of our nerves, but not as many as you would think.
Professor, in the tight junctions, those proteins, are they like possibly hydrophobic?
They don't, great question. They don't have to be hydrophobic because you're putting so many of them in a row. They basically, what they're doing, as the name indicates, they're just forming a really tight connection between the two cells so that it stops, it occludes the water from sneaking between them. Right? So that when you take that bath, you don't swell up like a sponge. When you're outside in the sun, you're not massively evaporating all of your water away. You're forming that waterproof seal to your skin to keep water inside, and keep water outside. So they don't have to be hydrophobic. There just has to be a lot of them clumped together in all these rows to limit that movement. Excellent. Great question. Any others? All right, if we are good with that, then let's talk about epithelial tissues. How are we on time? We're doing good. Okay, perfect. All right, so let's talk about the characteristics of epithelial tissues. So let's go back to our whiteboard. Now, the important thing to remember is that these are characteristics of all epithelial tissues. Does anybody know how many specific types there are?
I think 3.
If only. It's actually trickier than that. There are actually 8 specific types. 3 would be much, much easier, but there are 8 specific types. So obviously there's going to be a big difference between these 8 types. But these characteristics of all epithelial tissues are characteristics that all 8 types share. One of the first and most important characteristics of all epithelial tissues is that they are all very cell dense. Basically made up of cells and not much else. And these cells are not just randomly thrown together in a bucket. These cells are going to be arranged in layers, tightly packed layers. In fact, that's how we identify them. We identify the 8 types by the shapes, by the shape of the cells and the number of layers. Let's see how this works. A simple epithelial tissue is made up of 1 layer of cells. Whereas a stratified epithelial tissue is then made up of what?
Multiple.
Yeah, 2 or more layers of cells. So we have, as it turns out, 4 simple epithelial tissues and 4 stratified epithelial tissues. That's going to make things a little bit easier. So then the second way we identify our epithelial tissues, as I said, are the shapes of the cells. And there are 3 main shapes to epithelial cells. The first are broad, flat cells. Broad, flat cells look like they've been squished together. And in fact, as a result of them, we call them squamous. There are cells that are block shaped. And those block shaped cells are called cuboidal because they look like cubes. And the third shape are cells that are tall, narrow, and thin cells. And these, because they look like big, tall columns, are columnar. So let's do some math. If I have 1 layer of block-shaped cells, what type of epithelial tissue do I have?
Simple cuboidal.
There you go. If I have 2 layers of flat cells, what would that be?
Stratified.
Squamous.
There you go. See how easy that is? Put together the shape with the number of layers, and you have one of our 8 specific types of epithelial tissues. All right, so they're very cell dense. Let's pack some of this information a little closer together so I have more room to play with. And we identify our 8 types by the shape of the cells, by the number of layers that they have. Now, as we also talked about, all epithelial tissues line the surfaces of our body, inside and out. forming those barriers, forming that protection, allowing things to get in and out. And because they line the surfaces, one of the interesting characteristics is there is nothing above them. But obviously there can be tissues below them. What this tells us is that these tissues have a top and a bottom. You can't just take an epithelial tissue and flip it upside down and put it back on the body and have it work the same way. And of course, we need to have a fancy way of saying cells have a top and bottom, so we do. We say that epithelial tissues are polar. Oops. Right? When I think of polar, I think of like our ions where we had a positive and negative, but it's more like the Earth, or it's more like a magnet. A magnet is polar. Heck, this pen is polar. All polar means is that one side is not like the other side, right? One side has the part that writes and the other has the clicky things. This pen has some polarity. And if I'm taking notes, does it matter which way I hold the pen?
Yes.
Yeah, absolutely. So it's polar. And it's the same thing as here as well. There's a top to the tissue and there's a bottom to the tissue. And of course, because anatomists love to name everything, there are names to these. So let's do this. Here I have a tissue. This tissue happens to be a single layer of tall, narrow cells. So how would I identify this particular epithelial tissue?
Simple.
Simple columnar.
Simple columnar, perfect. So it is polar, meaning there is a top, but again, we're not going to get away with terms like top. In this case, the top is what is called the apical surface. And the bottom is what is known as the basal surface. So it's polar. The top and the bottom are different. The top is the apical surface. The bottom is the basal surface. Now, these things are superficial. So what is above the apical surface?
Air.
Yeah, well, air or something like that, or essentially nothing. Right? There's nothing above. We give that space above, essentially it's a space, and that space we give a name. So there's always a free space above an epithelial tissue. Now, notice my hand has an apical surface with a free space above it that is air. But what if I roll an epithelial tissue up into a tube? Notice the epithelial tissue still has a free space, but now it's an enclosed free space. And do we have a fancy name for an enclosed free space? Well, I'm asking the question, so what's the obvious answer?
Yes.
What is the fancy name for an enclosed free space? What do you call the inside of a hose or an inside of a straw or the inside of the small intestine?
Tube.
Tube is the outer structure. What do you call the space inside?
Cavity.
Cavity is not a bad term, but there's a better one. Lumen. So notice, there you go. All lumens are free spaces. But are all free spaces lumens?
No.
No, right? This free space above my arm is not a lumen. However, the free space inside of my mouth is enclosed by my mouth, so it would be a lumen. Heck, if you think about it, your entire digestive system is a hollow tube from your mouth to your anus. And the space inside here is the lumen. And it would be lined by the apical layer of your digestive system. In fact, there's a really interesting thing about the lumen of your digestive system, right? I need something small. Here we go. Perfect. Paperclip. You guys see that?
Yep.
If I lay it on my hand, is it inside my body?
No.
What if I wrap my hand around it? Is it inside my body now?
No.
No. It may be surrounded by my body, but it's not inside my body. Because to be inside my body, I would have to push it through a membrane and get it inside of my body. So, if I hold this paperclip in my mouth, is it inside my body?
No.
If I closed my mouth around it, would it be inside my body?
No.
If I swallowed it, would it be inside my body?
No.
No. In fact, the lumen of your digestive system is actually considered the outside of your body. Well, they have a fancy name for it. They call it the topological exterior of the body. Now, hopefully, nobody is swallowing paper clips. However, there are plenty of little kids who swallow pennies.
Wait, I have a question.
I have an answer.
I'm not sure if you covered it, but so you were saying if you put it in your mouth, it's not inside your body. Could you explain again what would make it inside of your body?
It's exactly what I'm going to do right now. So let's take that penny. A little kid swallows a penny. What happens to that penny? Enters their mouth, enters their digestive system, and what happens to the penny?
Hopefully exits the other side.
Yeah, most of the time it comes out the other side. A little cleaner, or a little dirtier, depending on how you think of it, right? And that whole trip through the body, it never actually entered the body, right? Your body has no need of currency. There are no receptors for pennies. So that penny just passed through the lumen of that hollow tube. Now let's get back to your question because it's an excellent one. Instead, hopefully none of you had a penny for breakfast this morning, but many of you, like me, had a nice big cheeseburger for breakfast. And when you have that nice big cheeseburger for breakfast, there's the bun, there's the patty. Let's draw the other bun on top. Again, you spit it up, chewed it up, swallowed it down into your stomach. And once it's in your digestive system, your enzymes, your stomach, your small intestine, break it up into all of the small pieces. Those small pieces then pass through the membrane of your small intestine. And as it passes through the membrane of your small intestine, once it passes that membrane, it is now inside the body, right? You have to pass through a membrane to get inside the body. Just laying this pen on my arm doesn't get it in my body. Wrapping my hand around it doesn't get it in my body. I have to shove it through the membrane to get it into the body. And it's the same thing here. That cheeseburger gets digested, gets broken down, and as it gets broken down, it gets past the membrane into our body. But the penny doesn't. The penny passes right on through and technically never enters the body. So a lumen is a free space.
I've got a question.
Yes.
And I think I've already thought of what the answer was, but so like a piercing, technically it goes through a membrane, but then it's scar tissue. So technically it wouldn't be in the body, but it is at the beginning.
Yes, I would agree. So if you get your ear pierced, that's a great question. So you get an ear pierce. It penetrates through the skin and it goes through the body. So it is technically inside of the body, right? And if you pulled it out right away, it would heal over, closing up that gap and protect you from the outside world. However, you're right. If you leave that earring in long enough, then the skin heals around it. And as the skin heals around it, you now have a space through your ear that would be, the lumen of which would be outside your body, because it wouldn't pass, because you now have a membrane that protects you from it. Absolutely. Which is why when you get, you know, that big, huge earring, right, in your ear, eventually you can pop those things out and that hole remains, right? And it doesn't bleed or anything when you take it out, because it is healed around it. Yep, absolutely. Great question. Any others? All right, so let's get rid of my digestive system, because while this is useful information, it doesn't affect all of our epithelial tissues. So excellent. So there's nothing above the apical surface, but there is stuff below it. These line surfaces, so they sit on top of other tissues. And do we necessarily just want them sitting on top of the other tissues where they can flutter away?
No.
No, we want them attached to the tissues underneath. And if we want them to attach to the tissues that underneath them, then we need a little bit of glue. So let's cheat a little bit, put my basal surface right here. Down here, I'm going to put some other tissue. It could be a connective tissue, it could be muscle tissue. Heck, it could actually be another epithelial tissue. But we have some type of tissue here. And we want to make sure that they attach. The way we make sure that we attach is by having a glue that connects the two together. So on the basal surface is the glue that holds the epithelial tissue to the tissue underneath. Now, are we going to get away with a term as simple as glue?
Mm-mm.
No, it has to have a name to it. And the name to that is the basement membrane, or it is also known as the basal lamina. Basal meaning bottom, lamina meaning layer. So either of those terms are acceptable. Basement membrane, basal lamina mean the exact same thing. Right? Basically, it is a glue. And again, even glue is a kind of a loose term. It is an extracellular, there's no cells in it, material or matrix. Now, when you and your sibling are playing football in the house and you knock over mom's vase and it breaks and you glue it back together, if you do a good job gluing it, do you see the glue?
No.
No. With one exception, in all of 430 and 431, you are never going to see the glue. So what's going to happen on the exam is I'm going to show you an epithelial tissue. I'm going to draw an arrow down here, and there's basically two questions I could ask. Question one would be to identify the surface or the side of the tissue. And what's your answer to that going to be?
Basal surface.
Basal surface. Or I'm going to ask you to identify the extracellular material that would be found in this location.
Basal lamina.
Right, or basement membrane. Right, so that is how I'll ask the questions. There's only one example in the entire 2 semesters where you're not going to see it. So most of the time, you do not see the basal lamina. So I'll ask that question that way. All right, identify the extracellular material found in this location. So that is how I will ask that question. And your answer to that will be basement membrane or basal lamina.
If you ask that question, would there be some other indicator on that cell? Like,
Well, what you'll be able to do is you'll be seeing that I'm pointing to the bottom of an epithelial tissue. Are we going to be able to tell the difference between the epithelial tissue and the connective tissue it's sitting on top of? Absolutely. So let's do that. We haven't gotten to our tissues yet, but let's find a really simple example. Come on, I want that picture. Give me that picture. So, like you, when you're studying this, I can go to the almighty Google. And when I go to the almighty Google, I can find something like this. And this will be what your exam is going to be like. There would be a picture like this. There will be an arrow like this. And question 1 could be, identify the tissue type. And what would your answer to that be?
single columnar cell?
Not single columnar, but
Or simple columnar.
simple
Sorry.
columnar. Excellent. I could point up here and ask you to identify the surface.
Apical surface.
Identify the space.
Free space.
Free space.
Free space. Excellent. Identify the surface. Just hold on. Stop it.
Basal surface.
Basal surface. Identify the extracellular material you would find in this location.
Basal membrane.
Yeah. Basal membrane, basal lamina, basement membrane, any of those are acceptable. Notice, again, you know nothing about anything else for this, but you can clearly tell the difference between this tissue here and the tissue below it. You may not know what the tissue below it is, but you can tell these two tissues look very, very different from each other. So we can tell the bottom of the epithelial tissue is usually pretty easy to see because you typically go from a cell that is very tightly packed with cells to a cell that is very loosely packed with cells. So it's pretty easy to find the bottom of an epithelial tissue. And of course, the bottom of the epithelial tissue is going to be the basal surface. And that basal surface is going to be where you find the glue. But notice they did a good job with this one. We don't see the glue. Like I said, there's only one example in all of anatomy and physiology that you will see the glue. Most of the time, we do a good job with the glue, and mom doesn't notice that the vase is broken. And that's the goal.
Professor?
So I can't, yeah, give me a second.
Oh,
I can't
sorry.
point here and say, identify this substance, because you can't see the substance. So that's why I'll word the question the way I did. Now go ahead and ask your question.
Yeah, I was just wondering, the bottom layer, is that a smooth muscle?
Nope, it's actually a real connective tissue. Yep.
Okay, thank you.
All righty, excellent. Oh, and since we're here, one more. Identify the organelle.
Nucleus.
Nucleus. Identify the organelle.
Microvilli.
Yeah, again, that's a little tricky because it's not the best view of it, but notice there is a little bit of fuzziness on the top, which again, just leads, as I mentioned before, to that key with polarity, because it's quite possible that there could be cilia or there could be microvilli on one side of this, on the apical side with this. So again, if I took this tissue and flipped it around upside down, it would definitely change its function, right? So absolutely, so there's that polarity, the apical surface and the basal surface. All right. We're not done with the characteristics, but what about these? Any questions on these? All right, there's two more really important characteristics we need to know about epithelial tissues. As I mentioned, epithelial tissues are very cell dense. And because they're very cell dense, there's not a lot of room for a lot of other stuff. Now, I know you haven't thought of it in these terms, but I know you know this, that all epithelial tissues are avascular. What does that mean? What does it mean to be avascular?
But they don't have blood vessels?
Yeah. What happens when I have an itch on my arm and I scratch it? Does blood come gushing out of my arm as a result of that?
No.
No. Why not? Because epithelial tissues have no blood vessels in them. No direct blood supply. So they are avascular. That doesn't mean they're dead. In fact, the opposite is true. Epithelial tissues are highly mitotic, meaning they divide very, very rapidly. And again, you haven't thought of it in these terms, but you know of that, because how many of those dead skin cells do you lose a day?
Millions,
And so how many new ones do you make?
millions.
Billions, right? When you burn the inside of your mouth by taking a sip of your coffee in the morning that's too hot, or taking a bite of that Hot Pocket before it's had a chance to cool, and you burn the roof of your mouth, how long until that heals?
A few days.
Yeah, it's only a couple days. In fact, in your small intestine, the epithelial tissues in your small intestine only live about 3 days. Every 3 days, you're replacing the epithelial tissues of your small intestine. So these are cells that divide very, very rapidly, but they don't have a direct blood supply. Essentially, what happens is the blood vessels will come up into like the connective tissues or the other tissues next to it, will come close to it so it can give it the oxygen, give it the nutrients it needs, but there is no direct blood supply to our epithelial tissues. So just because they're avascular doesn't mean they're dead. They're opposite. They're highly mitotic. And while there are no blood vessels in our epithelial tissues, most epithelial tissues are highly innervated. What does that mean?
Things like go in and out of them easily or quickly, or
What kind of things?
the oxygen and nutrients and water.
Not a
water,
bad
but
guess. They are very active that way, but that's not what innervated means. What does it mean to be innervated?
Found.
As a
Lots of places.
neuroscientist, a little part of me is dying on the inside. Dahlia, what were you going to say?
It's found in a lot of places, or
No. What does it mean to be innervated?
Is it highly sensitive to nerves?
Yeah, highly innervated means that it has lots of nerves in it. Right? Great example. How about your skin? The surface of your body is comprised of an epithelial tissue as well as some other tissues. And you are getting a fire hydrant's worth of information coming in from your skin. So much information coming in from your skin that most of the time you're not even paying any attention to it, right? Until someone like me points it out, you're not thinking about the feel of your socks on your feet or your chair on your back or your butt or the feel of your shirt on your shoulders, right? Those nerves in your skin are constantly telling you, hey, something's touching me. Hey, something's touching me. I'm feeling pressure. And most of that stuff you're ignoring most of the time because you're listening to me or paying attention to something else or eating a sandwich. It isn't until like someone like me points it out that you're like, oh yeah, I can feel the chair on my back. I can feel my socks on my feet, right? We receive a massive amount of information about the world around us from these heavily innervated epithelial tissues. Tremendous amount of sensory information, touch, temperature, tickle, pain, itch, all of those and more from these massive numbers of nerves giving us this information from our epithelial tissues. That's why it hurts when you drink that hot coffee and it burns the inside of your mouth, right? Or eat that spicy tortilla, right? That spicy hot sauce, which is painful both coming in and coming out because of all those nerves associated with our epithelial tissues. All right. Questions on this? All right, let's look at the handout from the lectures, make sure we got it all. Ranged in sheets, simple and stratified. Polar, apical surface and a basal surface. Apical surface has the free space above it. Notice here, we also see some examples of, for instance, some cilia or microvilli that can be on those apical surfaces. On the basal surface, notice we have that little bit of glue, that basement membrane or basal lamina, both of those are acceptable names for it, that are found down there. Very densely packed with cells. So not room for a lot of blood vessels. All of our epithelial tissues are avascular, but heavily innervated, right? They're forming our boundaries, so not surprisingly, many cell junctions. In fact, notice you can see our simple illustration here is kind of giving us some representations of some desmosomes holding these cell layers together. And like we said, highly mitotic. Perfect. Questions on that? Again, these are characteristics of all 8 specific types of epithelial tissues. So all epithelial tissues have these characteristics. But are there going to be characteristics that are going to be different between epithelial tissues? Specific types? Yeah. So now that we know what all epithelial tissues have, let's talk about individual ones. Starting simple, right? How do we define a simple epithelial tissue again?
One layer.
Excellent. One layer of cells. And we identify them by their shape. So as I mentioned, there are 4 specific types of simple epithelial cells, and we should be able to figure that out by putting them together, putting some names together. What would be one example of a type of a simple epithelial tissue?
Simple squamous.
Right, which is what? Define it. So now you've given me the name, define it.
Single layer of flat cells.
Excellent.
Flat epithelial tissue cells.
Yeah, well, yeah, we're talking about epithelial tissue, so that's fine with that.
You said specific, so I just want.
No, I appreciate that. Give me another one.
Simple cuboidal.
And define it.
A single layer of cube-shaped cells.
And what else?
Columnar,
Which you would define as?
one layer of tall, narrow, thin structures, cells.
Yeah, perfect. Absolutely. Excellent. Right, perfect. We've done that. That was super simple. And let's look at the pretty picture that goes along with it. Here we see an example of a simple squamous epithelium. Here we see an example of a simple cuboidal. And here we see an example of a simple columnar. Let's talk about each one of these. Obviously, if you think about it, simple squamous is the thinnest possible epithelial tissue, a single layer of flat cells. Is that going to provide a lot of protection?
No.
No. In fact, all of these simple epithelial tissues, even a single layer of tall cells, not very protective. So what's the advantage of having thin layers?
Diffusion or having things pass through it.
Yeah, better for movement in and out. Simple squamous is the classic example. One of the common places where you find this simple squamous epithelial tissue is in the alveoli in the lungs. In the alveoli of our lungs is where our gas exchange takes place. And what gas is, really we should say gas exchange, what gases are involved in gas exchange?
Oxygen and CO2.
Excellent. How long can you survive without oxygen?
A couple minutes at the most.
Yeah.
3 minutes.
How long can you let CO2 build up in your lungs?
Few seconds, 10, 20.
Yeah,
Well, however long you can hold your breath.
Right, exactly, right? A lot of times, if, again, someone's underwater, drowning, trying to hold their breath, eventually they're forced to breathe. Their body forces them to breathe. And most of the time, their body's forcing them to breathe, not because they've ran out of oxygen, but because they've built up too much CO2. CO2 is actually more important for determining our rate of breathing. So yeah, however long you can hold your breath, that's how long you can stand having CO2 build up in your lungs. So notice these are vitally, vitally important to get in and out of our body, but they are entirely passive. What does it mean to be a passive process again?
Doesn't require ATP or energy.
No ATP. So as vital as it is to get oxygen in, and as vital as it is to get CO2 out, we don't use any ATP to do that. It's strictly passive. And remember, when we talked about diffusion, one of the things that makes diffusion occur more rapidly is short distances. It doesn't get much shorter than a simple squamous epithelial tissue, right? Huge surface area. The surface area of your lungs is almost the size of a small tennis court. So you have this massive surface area, very, very thin membrane, because we want to make it as easy as possible to get that oxygen in and that CO2 out. Now, obviously, that is going to be important for diffusion, for exchange of materials. The other primary place we find simple squamous is where we just need a tiny barrier. We need a wall, but we don't want that wall to take up a lot of space. So we'll actually find it in the kidney. We'll look at it there. There'll be other places inside the body where you just need a thin barrier. This is going to be your fella. So there's some other places besides the lungs, but the alveoli is one of the classic places you find it. Conversely, simple cuboidal, single layer of cube cells, is one of the most common types of epithelial tissue found in lots of places. But where we're going to see it most is it is the simple cuboidal that is used to make our glands and ducts. So we're going to see it a lot when we talk about glands. We're going to see it a lot when we talk about ducts and tubes and things like that. All right, it's commonly rolled up into tubes or ducts. So most of the tubes or ducts in the body are made up of the simple cuboidal. Simple columnar is a little bit more sophisticated. Notice it can have microvilli on the surface, but it doesn't have to. So keyword there being can. And our simple columnar basically is found in most of the digestive system. Not at the beginning, not in the mouth, not in the esophagus, not in the anus, but basically from the distal third of the esophagus to the rectum. So basically lining the majority of our digestive system is where you'll find this simple columnar epithelial tissue. You can see that's why that microvilli would be useful, because what does our digestive system do? It absorbs those nutrients for us. All right. Questions on that? All right. So there are our 4 simple epithelial tissues. Any questions on that?
Wait, I actually have one question. I'm not
Yeah.
sure if you said it, but you said for the columnar,
Mhm.
how it says found in most digestive systems. So it's only found in digestive systems, but only some of them, like it won't be
Correct.
found anywhere else.
So like I said, if as I mentioned, your digestive system is a hollow tube that goes from your mouth to your anus. Okay?
Mhm.
Now, as I just finished mentioning, at the very beginning of your digestive system, in your mouth, in your pharynx, and the very beginning of the esophagus, that is lined by a different type of tissue.
Okay.
Your anus is lined by a different type of tissue. But basically everything else in between. So your distal third of the esophagus, your stomach, your small intestine, and your large intestine to the rectum. All of that, so the middle portion, the majority of your digestive system is where you find the simple columnar. And it's the only place you find simple columnar. Which
Okay,
is
thank
good.
you.
So when you see, no, it's a great question, because when you see the simple columnar epithelial tissue, you know you're looking at an organ from the digestive system.
Okay.
Yep. All right. Great question. Any others? Any other questions on our 4 types of simple epithelial tissues? Oops.
Did you just say 4 types? Did I miss one?
I have said 4 types numerous times, haven't I?
Yep.
And yet there's only 3 here, aren't there?
Yep.
That's troubling. Because one of the things that I've mentioned to you numerous times, and will continue to mention to you numerous times, is that anatomists hate you. And there are many, many ways that they show this hate for you. And this is one of them. They set up a rule. All right, this is how you identify your simple epithelial tissues. Single layer cells, talk about the shape, and boom, you have one of your 4 main types. And so sure enough, we have simple squamous, simple cuboidal, and simple columnar. But then they like to throw you curveballs. And in this case, the curveball is a pseudostratified ciliated columnar epithelial tissue. Now, this is a big, huge, mouthful statement that you will come to love to learn and learn to love and spell. But like many of the big alphabet soup terms we have, let's break it down to its parts. What do you think it means to be ciliated?
Got cilia on the top, on the apical.
And remind me again what cilia do?
Move mucus along.
Yeah, move substances. And you're right, that substance is almost always mucus. Along the surface. Pseudo-stratified. What does pseudo mean?
False.
False, right? False. Like chemistry is a pseudoscience, right? It is a false science. It's not a real science. Nothing important has ever come from it. Pseudo means false. Well, in this case, this is a pseudo-stratified. So what this means is it's a false stratified. It looks like multiple layers of cells, but it isn't. Instead, it is made up of irregular shaped columns. Let's go back for a second. If I move this down, then I think I have room. Notice when we look at a simple columnar. Simple columnar epithelial tissue is usually pretty easy to identify because all the cells are the same length. And not only are all the cells the same length, but all of the nuclei kind of line up in a row. Do I still have that picture we were playing with earlier? Yeah, here. Notice all of the cells are for the most part very similar in size and length, and all the nuclei pretty much line up like ducks in a row. Right, there's a little bit of fluctuation, but some of that fluctuation could also be, ooh, here's a fun term. Artifact. Anybody know what artifact means? I know you're all thinking of Indiana Jones trying to steal the golden amulet or something like that. What an artifact means is it basically means something that is not there in the living tissue. Let's start easy. In a real simple columnar epithelial tissue, are the nuclei all perfectly purple like this?
No.
No.
No. So why do they show up all pretty and purple like this on my slide?
They've been dyed.
Yeah, I've added a chemical to it. I've dyed them to be able to rectify that. Sometimes if I'm working with tissue and I want to see something in particular, what I may actually do while I'm processing the tissue is take a needle and poke a big hole in the tissue, putting something that's there that's not normally there so that I can easily find the area that I wanted to study. And sometimes it's not even on purpose, right? Let's talk about how you make a slide like this. The way you make a slide like this is you cut a piece of tissue out of something that is alive or was alive. You then freeze that tissue. You then use this massive, incredibly sharp blade to cut off about a 40 micron section of that tissue. You then dip it in all these different chemicals. You then use a paintbrush to put it onto a slide. You use the paintbrush to move the tissue around to spread it out. You then put glue on it. You put a cover slip on top of it. That is a tremendous amount of modification to the tissue. And not only have you changed the color, but could you have moved this location of the cells or caused some other type of damage or something like that to it while you're doing all those things? Yeah, absolutely. So all of that stuff is called artifacts. And so some of the irregularity that you see here is some of that artifact, and some is it's not perfectly and precisely arranged. But compare that to this. Notice here, when we look at, and again, we can look at the illustration, but I like looking at the tissue itself. We can still very clearly see the basal surface, right? A dead giveaway, you are looking at a ciliated pseudostratified columnar epithelial tissue, are the massive cilia. Notice they're much, much bigger typically than the microvilli are. But notice also the locations of the nuclei. They don't all line up like ducks in a row. Some are higher, some are lower. So it kind of tricks you into thinking there are multiple cell layers here. But when I look at the top and I see the cilia on top, I know I'm being tricked. And so I know I'm looking at a ciliated pseudostratified columnar epithelial tissue. And to finish it off, there are primarily two locations where we find it. Anyone know what those two locations are?
Nasal.
Nasal cavity. Remember, we talked about being at that Justin Bieber concert and having all that snot run out your nose because those poor little cilia couldn't keep up with it. Where else?
Throat.
Although more specifically in the trachea. Right? Your trachea is your airway to the lungs. We actually have cilia there that work as a mucus elevator. Again, we want to capture dust and debris before that air gets to our lungs. So we have these cilia that move the mucus up to the back of our throat so we can spit it out or swallow it. But remember, we also talked about how these cilia are delicate. So let's say I'm a 2-pack-a-day smoker. All of that smoke I'm inhaling damages those cilia. And now as I build up mucus in my trachea, my cilia can't move them anymore. So as a chronic smoker, how do I have to move that mucus out of my trachea?
You cough and spit.
Yeah, that smoker's hack. That right, to get that out of there. It's one of the reasons why, if you're a chronic smoker, you have to be really, really careful when you get sick that you don't take a cough suppressant. Because now you take a cough suppressant because you're coughing because of your illness, but you're still smoking, so the cilia are still damaged. So now not only is that mucus building up in your airway, but now it's draining down into your lungs, dramatically increasing the likelihood of pneumonia, COPD, even lung cancer as a result of that. So of course, the best thing to do is A, not smoke. But if you do smoke, then be very, very careful about taking cough suppressants. All right. Questions on that? So now we've gotten our 4 simple epithelial tissues. Now, I think we're doing good on time, so we will go over this more in the next class, but I want to, since we've got it here, let's go ahead and cheat and look at this one here. This is that simple columnar epithelial tissue that we've been talking about. And notice, conveniently enough, on my handout here, I have a simple columnar epithelial tissue. Your job, using a pen or a pencil, you don't have to use colored pencils, you don't have to use color if you don't want to, black is fine, but I'm gonna draw this. So there's my basal surface, there's my apical surface. I'm going to draw my tall columnar cells. I'm gonna draw my nuclei. And because my drawing had it, I'm going to, I heard myself coughing like that. Draw some microvilli on the surface. All right. I saw there was some kind of weird looking tissue down underneath here, so maybe I'll draw something down here with the occasional nuclei just to show that I know there's some tissues here, because then it'll allow me to be able to label the things that we have talked about. So what I'm going to do is over here, I have a list of the material that I am responsible for knowing. So what I'm going to do, we'll talk about specimen in just a minute. Simple columnar epithelial tissue. So I'm going to point an arrow at this, or maybe to make it easier for myself, maybe I'll draw a bracket. So here's a bracket. And then over here, I'm going to write simple columnar epithelial tissue. Probably not that big. All right. Then I'm going to see the next thing. We'll worry about membranes in just a minute. I know that this up here is my free space. However, notice it also says lumen. How do I know that this is a lumen?
Because the apical.
True, but are all apical surfaces facing lumens?
No.
No. But what do I know about simple columnar epithelial tissues?
Nothing's on top of them.
Well, I know that's true for all epithelial tissues, not just simple columnar. What do I know about simple columnar epithelial tissues? Where do you find them?
That in your digestive system is just a hollow tube.
Esophagus, stomach, small intestine, large intestine. Notice those are all hollow tubed organs. So since they're all hollow tubed organs, right, then I know it must be a lumen. Now, if you weren't sure, you are absolutely positively allowed to just write free space, because free space is always going to be correct for the space. But if you wanted to be tricky, you could also write lumen as well. right? I'm going to put an arrow that here, and I'm going to write nucleus, and so on and so forth. And that is what I expect. I do not want you putting a number here and a number here. I do not want you drawing an arrow from this to your drawing. I want you to write out your labels, right? So that if you needed to, if you wanted to, you could cut this out and just use this piece of paper as one of your flashcards when you were studying this. Okay? Notice not only am I going to label the things on here, but notice what I've put here are the key terms that I will use in identifying these things. So on the exam, I will show you this picture, and one possible question I could ask you would be to identify the tissue type. And your answer to that would be?
Simple columnar epithelial tissue, I mean.
Yeah, don't forget the epithelial tissue. Identify the organelle.
Nucleus.
Identify the side.
Apical.
Apical.
Identify the space.
Free space.
Identify the specimen in the field of view.
Stomach.
How do you know?
Because there's villi and macrovilli, or cilia, and, or no, there's macrovilli.
True, but guess what? There's microvilli in the small intestine as well. So what made you say stomach?
Well, my logic would say because of the stomach's, the acid, the hydrochloric acid in there, so it would have to be a little thicker. Not
Okay. I like that critical thinking. I thought maybe you had gone a little easier than that because on this handout, I have the word specimen sitting right next to stomach. Right, so if I ask for specimen, stomach would be an answer. Now, here's where things get a little tricky. On this slide, as it turns out, this one isn't actually the stomach, although it could be. I know for a fact that it's not because I'm the one who found it. But this particular image doesn't have anything that is a dead giveaway that it is the stomach. So when it doesn't have a dead giveaway, I will take that specimen question and turn it around. Instead, what I will do is I will say, identify one location where you, or organ, where you would find this tissue. And if I ask this question instead, what would a possible correct answer be?
stomach.
Stomach. Any
Large
others?
intestine.
Large intestine, small intestine. Yeah, exactly. And even the distal part of the
Colon?
esophagus.
Well, I guess
Colon-
colon is technically the large intestine.
Colon is part of the large intestine, but so is the rectum. Rectum would be too far. If you get to the rectum, then you've gone too far. Don't get to the anus because that's where it's different. But anything up from there, rectum, colon, those would be fine. But again, just saying large intestine would be okay as well. Right, so it's just another way of asking that specimen. Some things, as we'll see, and we will go through this histology together, when we look at these things together, there are some things that are a dead giveaway. You know exactly what tissue you're looking at. And then I'll just say, what is the specimen? There is actually nothing on this that tells you exactly what it is, but we recognize this is a simple columnar epithelial tissue. We know there's only certain places where you find simple columnar epithelial tissue. So instead, I would ask you that question, and you would say, I would say one location where you'd find this tissue, and you'd say stomach. The same way, if we go back here, if I showed you this picture on an exam and asked you one location where you would find this tissue, what would your answer to that be?
Nasal or trachea.
Yeah, nasal cavity would be fine,
or
trachea
trachea.
would be fine. Both of those would be acceptable answers. And again, if I ask for one, you only have to give me one of those two. So either of those would be acceptable answers. All right. So that is how this game is going to be played. That is why this handout is so important. because this not only is your an assignment that you're going to be turning in where you're going to be drawing these things and labeling these things, but this is your study guide for the epithelial tissues and connective tissues that you need to know histologically. All right. Like I said, we will go over this together. I don't know if we'll have time to go over all of it together, but we will go through a lot of it together, and we will start that tomorrow. All right. Questions on that?
Nada.
All right, excellent. I encourage you to use the resources like your histology atlas to get a head start at these things, looking at some of these things and identifying some of these things. Again, your atlas, your histology atlas has a tissues section that you can look at, but also remember, notice if you're looking at the kidney, then go to the part where it talks about the urinary system, look at the kidney. Yes, your book shows simple columnar epithelial tissues, but you can also go to the digestive system where it talks about the stomach. You can look at your ciliated pseudostratified columnar in the tissues, but you can also go to the respiratory system where it's in the nasal cavity and the trachea. So again, the other advantage of having these specimens is it tells you another location in your histology atlas where you can go to see these specific tissues. And notice, not only are we going to do this for all, these are all the histology tissues that you're responsible for histologically on the exam, but we're going to do the exact same thing with connective tissues as well. So that's our game plan for tomorrow. Tomorrow, we are going to hit the microscope hard, going through these things and talking about these tissues and everything that goes along with that. All right. Oops, I wanted to close both of those. Questions on that?
Not on that, but I was going to ask if you've heard anything from the bookstore, because I haven't gotten any update on like the ebook bundle, and I'm just worried because I think our 2 weeks is about to come up. I sent them an email, but I just wasn't sure if you'd heard anything else.
I have not heard anything, but I will look into it as well. Is anybody else still lacking stuff from the bookstore?
Just me? Okay.
Maybe just you, or people could also be asleep. I think people also turn on their cameras and then are paying attention to other things, you know, playing Minecraft or doing.
No, I did. I called them and canceled it because then I bought it online. But yeah, they told me the same thing.
Okay, yeah, and so again, I will check with them as well, but yeah, that is an option. Again, you do have the ability. Remember, with that you do have the 2 free weeks that has the book and all of that, and if you like that, you can just purchase it through them. You don't get an atlas that way, but there's also a lot of photographic atlases and things like that, the personal anatomy lab and all those kind of things that can supplement it. It's not as ideal as having it. I like the atlases, I like having the physical copy, but a lot of it is your learning style. So find whatever learning style works for you. But yeah, hopefully if I know they've done in the past, and that's why I'll contact the bookstore. If the bookstore is being delayed, then I can often talk to the Pearson rep, and they are able to either give us a new code or prolong your access for the free access for another week or so. So I know that is something that may be an option as well. So if you're not having your stuff for anybody who hasn't wanted to speak up, email me. I will check up on these things and I'll let you guys know what I find out. All right. Any other questions? All right, excellent. Well, then that is all the information that I have for you for today. Hopefully you've got a good game plan on what to get started on for tomorrow. And like I said, tomorrow we hit the microscopes hard. So we've already started looking at it. We will go more in depth on the next class. All right, I will see you guys then. Enjoy the rest of your day, study hard, and I'll see you tomorrow morning.
Thank you.
Thank you.
Thank you.
Thank you.